Okay, everybody, we're going to go ahead and get started. So we have a very packed agenda today, which is a great situation to be in. I'm not going to say problem to have. It's a great situation to be in. We have an unprecedented number of public comments that have been submitted. So I'm going to go ahead and just show you an overview of the summary of the public comments. Let me go ahead and share my screen so that folks online can also see. And so we had a total of 19 public comments received. There's actually some overlap between these, but we have 7 written public comments that you can access on our website. And then we have 12 oral public commenters who will be proceeding in rapid fire this morning. So I'm going to try and be stricter about time than I was yesterday, although we did pretty well. We still ended before 5 yesterday. But each of our oral public comments has been allotted 7 minutes plus 3 minutes Q&A. And so without further ado, I am going to turn it over to our first public commenter, who is Andrew Rowan from Wellbeing International. And let me pull up your slides, Andrew, and here you go. You can just click right here or on this arrow right here.
Okay. Thank you. Thank you very much, Nicole. And I'm really here to sort of talk about the past. I've been concerned that sometimes that the origins of ICVAM have sort of gone back into the mists of time, and that somebody needed to stand up and sort of explain how it came about. And so I wanted to do that. I was involved with this at the early days. Senator Proxmire was well known for being a gadfly on science. And one of his staffers was also very interested in the animal issue. And they started pestering the EPA on alternatives. What was the EPA doing on alternatives? The EPA sent a report to Senator about what they were doing. And he then asked for further information, asked for somebody from EPA to come. That became a bit of an embarrassment. And what ended up happening was that Proxmire added some funding to an appropriations bill, saying that he wanted money to go to Tufts, the Center for Environmental Management at Tufts, to look into the alternatives issue. I was at Tufts at the time. I was at the vet school. We had started the Center for Animals and Public Policy. And so I was the one who was then delegated to spend the money that the EPA had, or that Proxmire had advanced to the university. What we did was we established a dialogue group and included various animal NGO representatives and corporate representatives. J&J, Procter & Gamble, Exxon, Bristol Myers Squibb were amongst the corporations that were represented. We organized 3 workshops, one for NGO representatives, one for environmental, one for consumers, and one for government agencies. The government agency workshop was one of the toughest workshops I have ever chaired. Nobody wanted to be there. Warren's comment yesterday about everybody hated ICVAM, I saw the early stages of that. Nobody wanted to be at that workshop. They were there because I guess it was an EPA project. And we struggled through an afternoon of discussion. The dialogue group eventually decided to approach Congress, and this was a cooperative event with representatives from industry. Industry was very concerned that there was very little activity going on about what tests were validated and what tests were being required and so on, and felt that it was important to establish some agency or some effort to actually look at what the science was telling people. And so John Yam of Procter & Gamble and lobbyists from the MSPCA and the HSUS went up to Congress and persuaded Waxman to add language to the NIH Revitalization Act. That act was sort of in stasis during the Reagan years because of concerns about fetal research and abortion issues. But when Clinton became president, he signed it into law in 1993, and it had that paragraph, report language, that directed NIHS to establish ICVAM. Well, to establish a program, which then became ICVAM. And I was a member of the early SICATM, and you could see at the time that there was not a great deal of enthusiasm for this particular project. The NIHS were going through the motion, really didn't know what to do with this. And so I've been delighted to see that the NIHS has really sort of embraced it now. Part of the reason for that was that the Doris Day Animal League, Sarah Munson at Doris Day started pushing for an ICVAM authorization bill. And that was eventually passed in December of 2000, signed into law. And so now suddenly ICVAM was an authorized entity in the federal infrastructure. And that made, I think, a big difference to the whole process. But Dr. Birnbaum wasn't terribly enthusiastic about the ICVAM mission at the time, but she then started to change her opinions and was embracing this paradigm shift in toxicity in 2013. And that was about the same time that Warren Casey took over and Nicole Kleinstra became a central figure in all this stuff. And ICVAM really started cooking with gas. Finally, I'd like to just sort of comment on the fact that one of the things I've heard a lot about is that the development of alternatives is going to require more animals because you'd have to validate the alternatives against animal testing. When I've been tracking animal use over the years, and this is a graph showing animal use in each country that with the maximum set at 100. And so you can see that the mid-70s, that was the maximal use of animals in Great Britain, the USA 6 species, Netherlands, Switzerland, and Germany. The data is a little bit fuzzy in some places. The British data is great, but everybody else has its problems. But nonetheless, this is a rough indication of what's actually happened. So if you're looking at metrics, I was challenged on a webinar recently that, well, you say that animal use is going down. Well, how do you know? Well, the answer is that there is enough data out there to start making some real predictions about what's happened. And the fact that the chart looks so similar for these 5 countries, the US is, the green line is the total animal use, which is really sort of ILAR or ICLAS and ILAR surveys done up in the early 70s. And then the USDA numbers, there's just the 6 species numbers is the blue line. And you can see that the number, the shape of the graph is pretty much the same from one country to another. You've got the GMO mouse blip at the end there. In some cases, the Great Britain is a particular example, so is the Netherlands or Germany. But nonetheless, by and large, that's over now. The GMO mouse revolution is behind us, and the numbers are again beginning to come down. So just a very brief sort of outline of how ICVAM came into effect. I will note, by the way, that the dialogue group struggled to come up with any sort of coherent strategy and approach until Bob Scala of Exxon said that, you know, he was sitting in this group and he's saying, you know, if my management could see me now, they'd be horrified. And everybody says, well, if our management could see us now, they'd be horrified. So everybody in the room had the same response, was their management would be horrified that they're sitting around talking to one another. And that was the glue. It was amazing. At that point in time, everybody said, okay, well, what can we do? How can we make this work? And so the suggestion was that the federal government should do something to evaluate and validate new methods. And that's the genesis of it now. Thank you.
We have a few minutes for questions. Are there any questions in the room? Wonderful, thank you. Okay, so our next public commenter is Sally Thompson-Iritani from the 3Rs Collaborative, and she does not have slides, so I will just ask our AV folks to highlight our video for the folks on Zoom. Please go ahead, Sally.
Yes, thank you very much. And thank you for letting the 3Rs Collaborative join you here today.
Stop. Oh, shit.
Oh, okay.
Oh, he took it away. Good.
I really appreciate Andrew's summary there. And just like I just got to meet Andrew in person yesterday and so much respect and admiration for all the work that he's accomplished. So I just wanted to mention that. So I am Sally Thompson-Iritani, and just going to read the statement actually which was submitted on behalf of the 3Rs Collaborative. Wanted to be sure just to reiterate the efforts that the 3Rs Collaborative is making in these areas. We again appreciate the opportunity to provide written and oral input on the ICVAM related activities. We are a US-based nonprofit whose mission is to advance better science for both people and animals. Through facilitating collaborative 3Rs efforts, forwarding the use of the 3Rs and coordinating efforts across all stakeholders is therefore a central part of our organization. ICVAM and the 3Rs Collaborative have multiple shared interests and goals that could serve as opportunities for potential future partnerships. Two areas of focus that may be of particular interest to ICVAM include our initiatives devoted to advancing microphysiological systems and artificial intelligence for preclinical safety and risk assessment. Similar to the broad range of stakeholder representation on ICVAM's work group, the 3RC prioritizes evidence-based decision-making by collecting input from the full range of interested parties. For example, our MPS initiative includes 96 members from 44 organizations, primary commercial developers, including a formal consortium agreement and active participation from the FDA's Center for Drug Evaluation and Research and an MOU with the IQ MPS organization. Additionally, our artificial intelligence initiative includes 21 members from 15 diverse organizations, including the EPA, FDA, NIH, NIEHS, and the USDA Animal Welfare Information Center. We would like to congratulate ICVAM members for a productive year of activities that are advancing use of the 3Rs, validating new test methods, and coordinating efforts related to alternative methods across U.S. federal agencies. To build on this momentum, the 3RC would like to provide 3 key commendations and recommendations for ICVAM to continue focusing on in the coming years. The first one is to continue to enhance collaborations between governmental and non-governmental agencies, such as nonprofits, especially those that are balanced in their work across all 3Rs, and encourage collaboration between various stakeholders, researchers using animal models, and NAMs, developers, and sectors, being academic organizations, industry, contract research organizations, regulators, developers, and IACUC members. The recommendation aligns with the NIH Advisory Committee to the Director NAMS Working Group final report that emphasizes the need for combinatorial NAMS for this effort to be effective. There is a need to ensure clear communication, expectations, and goals. For example, we commend and recommend continued or extended participation of ICVAM members with the 3Rs Collaborative, such as ad hoc directors on our board and in our MPS, AI, and other initiatives to promote this collaboration. To help better advance the scientifically supported use of non-animal models in regulatory submissions, we recommend that end users, regulators, and technology developers work together on development and validation of methods for specific contexts of use, considerations for standards and outcome measures for specific contexts of use, and to show advantages of replacements in regulatory submissions. We also recommend funding a national-based, US-based balanced 3Rs, which considers replacement, refinement, and reduction center whose focus is on harnessing the power of collaboration to increase broad implementation of all 3Rs using evidence-based techniques. The second thing is to continue to provide investments for the independent characterization and validation of NAMs, especially those that have been commercialized, to ensure these technologies are well characterized and validated for context of use. This may include projects under the NIH's Complementary Common Fund, which I know we're hearing about today, as well as projects directly with the 3Rs Collaborative, such as the FDA Collaborative on the Drug-Induced Liver Injury Project. We also recommend balanced, accurate messaging on non-animal models that neither overpromise nor holistically condemn animal research, as this can alienate and activate cognitive dissonance in scientists currently using animal models. Progress will be facilitated by all researchers working together to develop the best model possible to support scientific objective with ethical considerations for both animals and humans. Partnering with nonprofits such as the 3Rs Collaborative may be valuable in these efforts. Finally, we'd like to continue to endorse and invest in implementing scientifically justified NAMs using evidence-based techniques to create change by promoting positive attitudes, providing training on implementation, and assisting in the normalization and socialization of 3Rs progress. Supporting nonprofits such as the 3Rs Collaborative that provide educational materials and advocate for responsible inclusion and analysis of NAMs in partnership with an understanding of the in vivo human and animal models. In conclusion, the 3Rs Collaborative really appreciates the opportunity to give feedback on the ICBAGM activities over the past year. Stakeholder feedback is crucial to policymaking. There's a clear need for funding to focus on 3Rs research, reproducibility, translation, and dissemination. As scientists that strongly support the development and implementation of the 3Rs in biomedical research, we look forward to the next year of ICBAGM activities that advance our mutual goals. Thank you so much. Next time I will have slides, but really appreciate being able to be here and talk with all of you.
Thank you very much, Sally. Are there any questions in the room? Paul, please, if you could use the microphone in the center of the room, that would be great so our online folks can hear you.
Thank you.
Yeah, there's a button. Charles will help you. Thank you.
Thank you for that presentation. I'm Paul Locke. I'm a professor at the Johns Hopkins Bloomberg School of Public Health. I'd just like to ask you a yes or no question, and then I'd like to, get your commentary on it. So does the 3R collaboratives follow, as many in the literature are now following, the idea that we should be pushing replacement over the other 2Rs? If the answer to that is yes, could you explain some of the projects you're doing? If the answer is no, could you explain how the collaborative sorts through the 3Rs? And also, I know you're an expert in this field, although you don't take enough credit for that. So if you can't speak on behalf of the collaborative, I would love to get your opinion on those issues. Thank you.
Yeah, and I would, not want to speak on behalf of the collaborative without conferring with my colleagues. So I definitely can give you my personal opinion. I do think we have a strong emphasis on replacement. I think that is the ultimate goal of these things. I do think we take a lot of time to sort through all 3Rs and emphasize the support for all of them. I do not feel like we're yet at a point where we can totally replace the use of animals across the spectrum. And so this is why we really I feel like we need to focus on reduction and refinement additionally. I'm very focused on refinement because I really see the importance of it in moving us forward and making sure that when we do need to use animals to support these scientific objectives, we take the best care of them that we possibly can. And so resources across all 3Rs would be ideal. I understand the emphasis for replacement, and I think that that is ultimately the goal that we're working towards.
Thank you very much. So our next public commenter is Dr. Sean McCulloch from RTI International. And Sean will also be giving an oral comment, no slides.
Good morning, everyone. I'm Sean McCulloch. I'm a senior respiratory scientist and principal investigator at RTI International. And I'm speaking today on behalf of my over 6,000 colleagues at RTI International. I would like to thank ICBAGM for the opportunity to deliver this public comment today. For anyone that is not familiar with RTI International, we are a nonprofit research institution that is dedicated to improving the human condition through the development and deployment of translational science around the world. Our team includes in vitro, computational, and regulatory toxicologists, as well as a broad range of the researchers in related scientific disciplines. This dedicated team applies their expertise to advance the development and use of new approach methodologies in basic science and applied research. And what I'm going to talk about today is a little bit more specific than our previous two commenters, so bear with me. Inhalation is one of the three routes of exposure to potentially toxic materials, and many inhaled materials elicit their effects, either beneficial or adverse, in the respiratory tract. It is also the portal of entry for many systemic toxicants. The vast number of data-poor new and existing inhalable materials, including ambient pollutants, occupational and commercial chemicals, pharmaceuticals, and consumer products, precludes the feasibility of using traditional in vivo animal studies to evaluate the potential adverse effects of these exposures. This challenge is exponentially compounded by considering the evaluation of exposures to mixtures of different materials, repeated exposure scenarios, and range of responses within the diverse human population. The use of in vivo animal testing methods for respiratory toxicology also has significant limitations resulting from fundamental differences in anatomy and physiology of the respiratory tract between humans and other animals. Utilizing human-derived in vitro respiratory tract models has significant potential to overcome these challenges by advancing human relevance and throughput of respiratory toxicity testing, while also reducing reliance on in vivo animal testing. Unfortunately, the use of in vitro approaches for the evaluation of respiratory toxicity has lagged behind other tissues and routes of exposure due to several key technical challenges in recreating the unique conditions of the respiratory tract in vivo using in vitro models. I would like to highlight several of these considerations with the goals of raising awareness, encouraging dialogue across the governmental agencies and other stakeholders represented here today, and ultimately positioning the field to overcome these limitations to the advancement of alternative methods in respiratory toxicology. My comments today will be focused on in vitro respiratory models and assays. However, there are other critical aspects of in vitro respiratory toxicology testing that are equally in need of similar consideration. The highest profile case study involving in vitro respiratory tract modeling to date was conducted to evaluate the potential of the pesticide chlorothalonil to cause toxicity within the respiratory tract. This study evaluated the effects of chlorothalonil exposure on air-liquid interface, commonly referred to as ALI, primary nasal epithelial cell cultures. The in vitro data collection began in 2016, and the final report was issued by the Federal Insecticide, Fungicide, and Rodenticide Act Scientific Advisory Panel, commonly referred to as the FIFRA SAP, in 2018. Conducting this study provided a first step for the use of in vitro respiratory models in regulatory decision-making, as well as the opportunity for a diverse range of experts to provide feedback on ways in which in vitro approaches may be improved moving forward. In the 8 years since data collection began on the chlorothalonil study, research conducted by several laboratories, including my own now at RTI International, has demonstrated that respiratory tract cell types other than epithelial cells are both targets and mediators of the effects of inhaled toxicant exposures. Considering that the lung is composed of more than 40 known cell types, these findings are not unexpected. However, they highlight the need to expand the scope of respiratory toxicity testing beyond the epithelial cell-only paradigm. While the field has grown familiar with epithelial-only models, our collective understanding of respiratory biology indicates that epithelial cells are not the only cell type involved in lung structure and function or the effects of inhalation exposures. Translating this knowledge by investing in the development, validation, and availability of in vivo relevant multicellular models is key to ensuring human relevance and ultimately the successful use of in vitro models for respiratory toxicity testing. Regardless of the number of cell types they contain, differentiated primary in vitro respiratory tract models are generated using cells isolated from individual donors. Primary cell-based models provide considerably improved physiological relevance compared to their cell line-based counterparts. However, they are also generated from different donors and exhibit inter-individual variability. The capability of primary cell-based models to represent the biological diversity within the human population provides a level of real-world human relevance that cannot be achieved by traditional animal testing methods due to their reliance on inbred rodent strains. Unfortunately, the scope of inter-individual variability in vitro respiratory tract models is poorly understood. Characterizing this impact is critical for determining study sample size, considering sensitive and susceptible populations, and expanding the scope of in vitro model application to incorporate biological variables such as age, sex, race, exposure history, among others. It is also critical for ensuring that the field does not inadvertently develop new chemical testing and regulatory paradigms that are not representative and thus not equally protective of the diverse human population. Primary cell-based models also vary in the origins of key materials, as well as methods used for model construction and maintenance. Experimental exposure systems and endpoint assays used to evaluate the effects of exposures on in vitro respiratory epithelial models are similarly subject to the influence of a range of variables. Quality control criteria and reference standards are often absent across in vitro respiratory models and toxicity assays, and typically lack consistency and data-driven rationale when present. In vitro systems, assays, and exposure systems for respiratory toxicity testing have not been sufficiently validated to ensure the sensitivity and specificity, normal ranges of technical and biological variability, and performance relative to appropriate standards and controls. Further, the comparability of different cellular models and transferability of assays, and ultimately the reproducibility of study outcomes across laboratories over time remains untested. Thus, it remains unclear whether the results of key studies, such as the chlorothalonil case study or the 2,3-pentane-dione study presented yesterday, can be independently reproduced. In the absence of assessing their reproducibility, it is possible that they may have been the result of intrinsic properties of the specific donors that were used to generate the in vitro test systems, variations in cell culture maintenance or dosing methodology, differences in endpoint assay procedures or reagents, or uncharacterized variables. The cumulative impact of the absence of standardization, comparability evaluation, and validation is a significant limitation to the broad acceptance of in vitro alternative methods for respiratory toxicity by decision makers. While less well appreciated, these issues also challenge the rigor, reproducibility, and relevance of respiratory biology research. Addressing these knowledge gaps will require significant investments of expertise, thought, effort, and resources, but are absolutely necessary to build confidence, gain acceptance, and ensure the protection of human health. Fostering collaborations across laboratories and providing meaningful and sustained funding opportunities to support such evaluation and validation efforts is critical to ensuring successful integration of in vitro respiratory tract models and assays into respiratory toxicity research and testing. In conclusion, advancing and validating in vitro approaches for the evaluation of respiratory toxicity is critical to ensure the reliability, reproducibility, and human relevance of respiratory toxicity testing and research. Characterizing interindividual variability in primary cell-based in vitro models is also necessary to ensure that they ensure the representation of the range of responses across the diverse human population in respiratory toxicity testing, as well as basic science. Doing so provides an opportunity to leverage the ability of in vitro systems to inform on population diversity in respiratory toxicology where existing in vivo animal models cannot. In contrast, failing to characterize the impact of interindividual variability risks inadequately protecting human health and furthering public health inequality. The forthcoming NIH Complement Animal Research and Experimentation, or complementary, initiative will undoubtedly make key contributions to the advancement of alternative methods. However, it is just a first step. The scope of the challenge developing reliable, predictive, properly controlled, and transferable systems and assays in organ and tissue systems throughout the body is truly immense. At RTI International, our mission is to advance the human condition, and we are looking forward to the availability of more funding opportunities to allow collaboration with new and existing partners to build key infrastructure for the use of alternative methods to advance human health protection for applications in respiratory toxicology and across other tissues and routes of exposure. I'll be happy to discuss this topic further. If anybody is interested, whether it's questions or after the fact, please feel free to contact me. My email address is my last name @rti.org. Thank you.
Just to clarify, Sean's email address is smcculloch@rti.org, not my last name. Just kidding. I do have a quick question for you, Sean. What would your top recommendation be? I mean, we heard yesterday about the issues of commercial platforms that have only one donor, available to represent certain cell types in the lung. So what would your top recommendation be to increase the availability of more diverse donors for commercializing and operationalizing models?
That's a great question. And so the easiest answer is to support a repository for readily available materials. There is a repository that is NIH funded that has some materials available for respiratory model development called LungMap. However, those materials are without getting too technical, you basically get a slurry of cells and you have to isolate your cells of interest. So while they are accessible in one way, they are not as accessible as they could be in another way. So providing support funding perhaps for the development of a repository that has specific donor match cell types available for researchers, and realizing that there would be a lot of complication in this, even for potentially commercial developers, to have access and consistent access to, with consistent collection, expansion, and maintenance protocols would definitely be a big help there.
Okay. Thank you. Any other questions? We have one in the room.
Okay, thanks. So yeah, not sure if this is a comment or a question yet. I'll see how it comes out. So I see sort of two phases. The first part, you were talking about the challenges and limitations with the current animal tests, and then the in vitro models being available, having promise, and then additional work that we could do on those in vitro methods. So where do you see as that point of when we say, okay, we're as good as the animal test and we should continue to improve, but also acknowledging, well, the animal test doesn't give us inter-individual variability. So kind of where do you see those inflection points, or do you have any thoughts on that?
That's a really complex question.
Oh, hi, Amy Klippinger, PETA.
So it's a great question, and it's a really complicated one. And I think at the end of the day, the answer is we need more information, and the answer is likely going to be different across chemical classes, or physical chemical classes and exposure scenarios. And so I think we have to interpret the in vitro data interpret the data as they are. The data that are typically coming in today are in epithelial-only systems, and the respiratory epithelium plays a general role in the respiratory tract, but also has specialized roles in different regions of the respiratory tract. And so we need to interpret the results of those studies and really be conscious of what they can tell us and what they can't tell us, and how that may or may not be more human relevant than the traditional in vivo testing. And really, I think at this point, unfortunately, it's kind of a case-by-case basis until we build a sufficient database, if you will, or knowledge base to make more broad extrapolation into the applicability. But I think at the end of the day, the focus should really be on where we are is where we are, but we need to be looking forward to how do we implement, especially technology that is relatively well, as in vitro systems go. It's not, multicellular models haven't been published by one lab or two labs kind of thing. So these are things that are, and in some cases, I know the work that I did at EPA, we published our protocols, very detailed protocols on open source protocol repositories. So at least for the research side, those models can be reconstructed. And on the, for those that favor the use of commercial models, the two primary respiratory tract culture providers offer co-culture systems for at least the conducting airways, the nasal region and tracheobronchial region, and in some cases lower into the airway. So I think there's the opportunity to integrate those there and start really thinking about moving forward into how can we get more in vivo relevant models to get us closer to humans without relying on those animals.
Yeah, that makes sense. So keeping in mind when we get to the as good as, but then continuing to progress after that, I guess.
I mean,
Getting the
Yeah. Time. Justin, did you have a question, or are you coming up with it?
I did.
Okay, super quick.
Sorry, I'll try to make it quick. So Jessica Ryman, American Chemistry Council, ACC. I think you had mentioned basically variability based on the person and the need to have, information from multiple people in order to get a handle on the variability. I was just kind of wondering for like a common endpoint, what kind of variability you've seen so far? Like 1x, 2x, 3x, 10x, like what have you seen so far in practice?
I could talk through this entire meeting on that, but I won't, because Nicole will hook me off the stage and throw me out the back. But just to give an example, so we have a study that we're in the process of writing up where in response to a common ambient air pollutant and commercial chemical, in a donor group of 14 individuals with looking at cell viability as or culture viability as outcome, our range is from 0 to 100%. So just as an example, that variability does change based on the endpoint that you're looking at, based on this. And take this for what it's worth. It's a single study with a single toxicant. However, these types of studies, getting co-cultures from 14 donors and then doing concentration response studies across, in this case, I think we did 10 endpoints. That's a big study. It costs a lot of money. And so there aren't a lot of those studies out there to kind of get an idea of how representative that particular study may be, or what other studies may be out there to indicate how many donors do we need? Because I think at the end of the day, one of the key questions is, what do the data suggest in terms of sample size considerations? Do we need, is 3 donors enough? Is it 5? Is it 10? And how is that impacted by the endpoints that are being used for whatever the evaluation is? Happy to talk more about that.
I presume those were all healthy donors as well.
Those were all normal, healthy donors. Yes.
So disease conditions add additional complexity to that equation.
Absolutely.
Okay, thank you, Sean. So our next public commenter is Jessica Jessica Ryman from the American Chemistry Council. And do we have slides for you?
I do.
Yes.
Yeah.
Pull them up.
Okay, great. Thanks so much. So we should be ready to roll. So I am Jessica Ryman with the American Chemistry Council, ACC. I'm joined by my colleague Rick Becker, who is participating remotely. And of course, I'll be giving the presentation for today. So we would like to begin by applauding ICFAM for adopting a scientific confidence framework, or SCF. For the last, over a decade or so, there have been conversations within this community that, traditional round robin validation takes a lot of time, takes a lot of resources, and really isn't very practical. And so the conversation has, evolved to, well, we need to find a way to do, fit for purpose validation. But the question was, how do you do that? And within recent years, the idea of scientific confidence frameworks has come to the fore in order to operationalize or provide a way to do fit for purpose validation. So we applaud ICFAM for adopting a scientific confidence framework. We think that this should really accelerate the adoption and also evolution of new approach methods, because these things are, these NAMs are coming online and changing so fast. We need to be able to keep up with them. But in thinking about this, I think it's important to note that the regulatory science community is going to need training and encouragement to embrace scientific confidence frameworks. I mean, people over the last couple of decades have been socialized basically to expect lists, right? It's like, show me list of NAMs, tell me what I can use them for and how I need to do it, right? And so a scientific confidence framework is a real change. It's a real shift from that. And so it's probably going to take a lot of education to get people to understand them and to embrace them. And then, one day as scientific confidence frameworks come online, it's even possible to envision that lists may one day be on their way out, as scientific confidence frameworks are increasingly being phased in. So that's a sea change. And speaking of other sea changes that we have going on in the regulatory scientific community, we also have a situation where artificial intelligence, or AI, is starting to converge with high throughput, high content, data-rich toxicology, right? And so there was a publication earlier in the year, if you haven't seen it, written by Nicole Kleinstruer and Thomas Hartung, that described basically this convergence of tox and AI as the end of toxicology as we know it. And we think this is completely plausible because it's already happening. You see in the drug development space, we see the conversion of AI and toxicology. We had a lot of AI-related tox posters and talks at SOT in Utah just this past spring. And so really, this could be as big as or as big of a shift as Tax 21 was almost 20 years ago now, right? And Tax 21, of course, is in large part and partially, well, there's also the ICFAM Act, but Tax 21 is a big part of the reason why we're here today. And so I think we think the lesson from Tax 21 or Tax in the 21st century is that this convergence of Tax and AI is really going to take the involvement of the entire regulatory scientific community. So that's it's academe, it's the federal government, it's industry, it's NGOs, it's nonprofit. It's going to take all of us. And so we ask ICFAM to identify opportunities for meaningful engagement for all stakeholders in what we expect to be a very large transition in our field. And so I want to go on now and mention that, at ACC, we don't just talk about NAMs, we also fund NAMs research through our Long Range Research Initiative, or LRI, which is headed up by Rick Becker. And I have a picture of the front of the website there, as well as the link at the bottom. ACC's public comments are, I think, posted on the ICFAM website now. And at the end of our public comments is this slideshow. So you can get these slides pretty easily. But we found a lot of research. I'm not going to talk about all of it. I'm going to give an example just of some of it, okay? So in the area of improving chemical safety testing technologies and the design of fit-for-purpose assays and the advancement of data interpretation, we contribute to the consortium out of HESI for Amex for assessing signatures for integrated safety, or the OASIS consortium. We contribute to a TexVal toxicity chip testing consortium out of Texas A&M. And then at Cytovation, we're funding 2 projects. We're funding a fit-for-purpose, development of a fit-for-purpose in vitro model for lung toxicity, as well as application of toxicogenomics and next-generation risk assessment. And so these projects and more information about them, as well as, when the papers are published, because everything gets published, is present on our website. We have a whole research catalog of other projects there for over the years. And then finally, just to wrap up, we'd like to encourage ICFAM and everyone in the room and online to consider value of information, VOI analysis, is an additional consideration in the evaluation of the utility of adopting NAMs for regulatory use. I mentioned here that EPA published a VOI analysis in 2023 for its 5-day ETAP assay compared to traditional human health risk assessment. And that's a 5-day study that's done in animals looking at transcription. And I think Allison Harrell gave us a really excellent presentation yesterday, much more in depth than this, on EPA's VOI analysis for the ETAP. I'll just note that in 2024, we published a VOI analysis that looked at ETAP and traditional human health risk assessment, but we also looked at the threshold for toxicologic concern and basically found a higher VOI for TTC than for the other 2. And so really VOI can be a really useful means for informing tiered testing frameworks in terms of progression. For example, TTC, then ETAP, then longer duration in vivo studies. So that concludes my presentation, and I'll be happy to answer any questions that you have.
Any questions in the room for Jessica? And nothing online. Okay, thank you very much. Okay, so we will now move to our next commenter, who is Dr. Paul Whaley. Let me just sort out the slides on the side.
Okay, thanks, Nicole. While you're doing that, can I just check that everyone can hear me okay?
Yes, we can hear you just
Fantastic.
fine.
Super. So
And
potentially a bit of construction noise in the background, but there isn't any right now, so hopefully that'll stay away for the next 5 minutes or so.
we do not
Good,
hear.
I think we're good to go.
Yeah.
So, Nicole, if you could advance the slides for me, that would be fantastic.
Of
So-
course. Let me know.
Super. Right, so thank you, everyone, and to ICFAM for giving me the opportunity to make this public statement about the important role that publishers have in supporting the regulatory application of scientific research and a new initiative we're undertaking to promote the reproducibility of NAM studies. So my name is Paul Whaley, and I'm the editor-in-chief of Evidence-Based Toxicology. This is a new journal created to support open science innovation in toxicology and environmental health. And it's the official journal of the Evidence-Based Toxicology Collaboration at Johns Hopkins Bloomberg School of Public Health. Our editorial policies at EBT include open peer review to improve the transparency of decision-making on submissions, high standards for data availability, the use of preprints to prevent publication bias, and encouraging researchers to publish their planned methods before they collect their data. Next slide, please, Nicole. So today I'd like to draw your attention to a new initiative we're undertaking in collaboration with the Centre for Open Science to promote the reproducibility of research. This is the introduction of a novel type of manuscript format that we think may be of particular interest to researchers working with NAMs and those with an interest in the validation of NAM methods. We call this new submission format pre-registration templates. That's kind of a complicated name, but the idea is simple. It's a methods template for making a study reproducible, published in isolation of the study itself, with the sole intent of guiding another researcher through the procedures they need to go through to successfully plan a similar study. So if you wanted to, you could call this format study planning worksheets or SAP development templates or something equivalent. Next slide, please. So what happens in this format is that we can have researcher one on the left develops a method for doing a study. They conduct the study, maybe once or twice or 3 times or 10 or whatever. They find it works pretty well. They would like other researchers then to be able to use that method too. So what they do is they create a template for their method for another researcher to follow when planning their own similar study, publish that. Researcher 2 comes along, discovers the template through PubMed indexing or Web of Science or wherever. They use it for designing their own study, and they get better research outcomes as a result. These better outcomes can include a clear articulation of the research question or theory behind it, fuller description of materials being used in the study, clearer specification of the outcomes of interest, and a more complete account of how the collected data will be analysed, kind of all the stuff that is very desirable when we're trying to understand how good a study is. And then the completed template could also be pre-registered, demonstrating that methods were planned before the data was collected. And over time, the idea is that the publication of these templates creates a pool of peer-reviewed guides, the comprehensive planning of many different types of study, designed by their creators to be reusable, that any other researcher can follow to help them apply other people's methods in their own labs. Next slide, please. So besides being a pool of high quality methods, being a value in its own right, we think these templates are interesting for people working with the NAMs in relation to development uptake of this type of testing technology. So firstly, we think they could greatly lower the entry barrier to reusing NAM methods, because at the moment, using another researcher's approach often requires careful interpretation of the method sections of multiple study manuscripts. Each of these method sections only partially describes everything that needs to be done to make a study work. So a reusable peer-reviewed template that helps a researcher provide a complete specification of a method will reduce the amount of guesswork involved when one researcher attempts to use another approach. Secondly, we think they could help with improved compliance with research guidelines. So we know that general guidance, such as OECD's GIVIM document, can be challenging for researchers to interpret into specific study designs. So templates can help with this. They allow one team to share how they crack the problem of designing a particular type of study to be compliant with a given standard. Creating a template, this process solves a problem for the whole community, eliminating potentially redundant work by other teams having to independently crack the problem of interpreting that standard themselves. And thirdly, pre-registration templates could support the retrospective validation of NAM methods. should involve a move away from prospective approaches to methods validation, which are proving somewhat cumbersome for a field as complex and fast-moving as NAMs, and also to the increasing expectation that all studies relevant to chemical assessment be systematically evaluated on their own merits. Because pre-registration templates provide a mechanism for integrating comprehensive planning of a study, the complete documentation of its methods and results, decision makers should be more readily able to determine how confident they should be in template-guided studies when using them to inform policy. Next slide, please. So yeah, to make science work better for decision-making, publishers need to be proactively engaged with researchers and regulators. And we think our new pre-registration template submission format is a step in this direction. We welcome contributions to develop case studies of the approach and start building evidence base for its potential effectiveness and utility. If you're interested, and I hope you're interested, then please contact me for more information at p.whaley@lancaster.ac.uk. Thank you very much for your time and your interest.
Thank you, Paul. We'll open it up for questions and I will take the prerogative of asking the first question. Do you envision a sort of iterative feedback process built into these submissions? So having a pre-registration template for a protocol gets published, and then people who are trying to use that protocol would have sort of a forum or an ability to provide feedback on any of the challenges that they've encountered or opportunities for improvement or questions?
Yeah, so so the technology for templating is still kind of in its early days, but I think, good templates would utilize maybe something like the protocols.io protocol development platform. The platform's available. And if we received a submission that was created using that platform, then we would certainly be able to leverage the forking and versioning and commenting feature of that platform to facilitate that feedback process, I think. This would be very much where we try to integrate our journal submission review workflows with the options and opportunities that the different technological implementation of these protocol development platforms would provide as well.
Okay, great. Thank you. Are there any questions in the room? Not seeing any. Are there questions online? Yeah, Emily, please go ahead.
Actually, now we have two. So first question, how is this different than current protocol repositories? And do you have an SME helping to develop the protocol templates?
So the templates they're a bit like, well, they're not quite protocol. So protocol is the plan for a specific study. Template is a worksheet that helps somebody do a good, comprehensive, fully reported plan. So there's a difference between what you would need to know in order to do a specific assay versus some general template, maybe that related to like a set of assays that take you through the things that you should think about in terms of what needs to be anticipated in order to provide a good plan, right? So it is definitely intended as a template or a worksheet or a structured guide to creating a protocol rather than being a protocol in and of its own right. But you could derive a template from a high quality protocol because the high quality protocol will be an instance of a good study plan.
All right, thank you. And then the next one, are there plans to align this initiative with the current CUSP CUSP project through FDP?
I think we would need to look at that. This is a new idea. This project that's being referred to here is a new thing. So if someone wants to just drop me a note about that, then I'd be very willing to look at that and discuss it.
Okay, great. Thank you so much, Paul. All right, so our next public commenter is Dr. Amy Clippinger from the PETA Science Consortium International. Amy, do you have slides? No slides, okay. So I'll just ask our AV, let me stop sharing and then ask our AV folks to highlight our video. Great, thank you so much.
All right, great. Well, good morning, everybody who's here. As Nicole said, my name is Amy Clippinger. I head up the regulatory toxicology work at PETA. Really enjoyed hearing the presentations yesterday and looking forward to hearing more today. And thanks again to Warren for that reminder that this is the 10-year anniversary. I can't believe it's been that long, but it's really great to see the attendance grow and really the progress and the scientific advancements over the past decade. So as I was listening to the conversations yesterday, what really stands out to me is that there are a lot of people working, collaborating, a lot of projects ongoing to advance the use of reliable and relevant methods, and many opportunities today where these can be implemented. One place where this was recently demonstrated was with the January EPA Office of Pollution Prevention and Toxics publication of its irritation decision framework. This was a really great example of vetting methods based on their context of use, their reliability, their relevance to humans, as described in the ICVAM report on the validation, qualification, and regulatory acceptance of NAMs, which was mentioned yesterday. If anyone isn't familiar with that ICVAM report, maybe this is going to be mentioned later, but I think Nicole is giving a presentation on it this Thursday. So it's a really good read. And if you don't want to sit down and read it, Nicole, I'm sure will summarize it really nicely on Thursday in a webinar. So just I wanted to make sure I thanked NICEDM and ICVAM and all of the ICVAM member agencies for their leadership and support in developing that report. The other piece of the EPA's irritation decision framework that was really important was just simply putting this document online, making it publicly available to stakeholders. It's not always that we need the development of a new method, but sometimes just increasing clarity about the use of existing methods is really important so that companies can use those methods. So I look forward to seeing the publication of additional similar documents from EPA and other agencies that clarify how methods and data are considered and can be used to meet regulatory requirements. Two other places where I see the ICVAM report being useful are in the area of medical devices and sunscreen. As many of us know, a number of methods have been stalled in the FDA's medical device development tools program for a number of years, with a kind of ever moving and increasing higher bar to gain confidence in these methods. I mentioned one of these yesterday, the in vitro reconstructed human tissue model for assessing skin irritation potential of medical device extracts, which has already been extensively validated. It's included in ISO standard. It is accepted by the European Union member states, Australia, Japan, China, and so forth. So this should be an area where I think if we kind of all put our heads together and applied the principles of the ICVAM validation report, that we could get this method pretty readily accepted. The second area I mentioned is sunscreen. As many of you likely know, the FDA is asking for the generation of additional data to keep existing sunscreen products on the market. My team regularly hears concerns from the public that they do not want their sunscreen products tested on animals, and in particular, when the broader scientific community around the world does not agree that it's needed. So I encourage the key people from the FDA to engage in the ongoing conversations and efforts to develop and gain confidence in reliable and relevant non-animal approaches to assess these products. So overall, just to sum that up, I hope to see the application of the principles outlined in the recent ICVAM validation report to tackle today's issues, including for sunscreen, for skin irritation testing of devices, and well beyond that. And then while we're working towards these bigger, longer term, very important projects, to remember that there are shorter term milestones along the way, simply providing clarity in the acceptance of existing methods so that we're incorporating the best science as efficiently as possible. Thank you.
Are there any questions in the room? No. Online? No, okay. Okay, thank you. So our next presenter is Dr. Randolph Ashton from Neuroseta LLC. And Randy, I see you're online and you are sharing your slides. So hold on, let me just pull the Zoom over so that everyone else in the, hang on, that everyone else in the room can see. Oh, don't leave, Randy. He ran away. Oh, there he is. Okay.
No, I have a sick kid, so just trying to minimize noise.
I'm going to ask you to speak up just a teeny bit. Your audio is a little bit low.
Me now?
Yes, maybe just channel your inner loud voice.
Is this better?
Yeah.
Okay.
Okay, go ahead.
All right. Well, I want to thank everyone for the opportunity to present. The platform that I'm presenting today from Neurozetta is the Rosetta RAID platform we presented about a year ago, and I just wanted to provide an update. We think this is a transformative tool for quantitative high throughput screening of human developmental neurotoxicity. And is my audio level okay? Yes? Nicole, is the audio level okay?
You're muted, Nicole,
Okay, great.
but you sound great.
Okay,
I am
thanks.
aware. Yes, we can hear you in the room. It just took me forever to get to the audio, the mute thing.
Okay, no worries. All right. So today's current status of in vitro human developmental neurotoxicity testing is the in vitro battery that was proposed by the OECD. And so this is a diagram of human embryonic development in the central nervous system, brain and spinal cord, with some barriers that are being presented here in terms of where they occur. And then this is the current battery, sort of the list of assays that are within that battery. And a lot of these that you see in the early time points, these are human pluripotent stem cell derived assays, but they use progenitors in order to conduct the assay. You have a number of assays that are fetal cell derived assays, and then you have rat neuronal assays. And what's apparent from here is a glaring hole in the 0 to 6 week time point. And this is actually the time frame in which you have the initiation of events that cause congenital birth defects, such as neurotube defects, spina bifida, as well as the initiation of events that cause autism spectrum disorder. And so the importance of this time frame is such that these are so early in development, often before people know that they're pregnant, and they can be diagnosed pretty early. So neurotube defects are diagnosed within the second trimester. Autism spectrum disorder is now diagnosed within 18 to 24 months post-birth, and so they can be directly correlated to various chemical exposures. And so what we propose is the use of neural organoid platforms. And so this technology that has been really come to the forefront within the last 2 decades, where you can take human pluripotent stem cells, that be embryonic or iPS derived, culture them as aggregates, and then they start to undergo aspects of CNS morphogenesis, the process of forming brain and spinal cord tissues, in really an unprecedented manner as you guide them down the culture pathway. And so this is really great, a really powerful tool that is used in a lot of laboratories, including my own at the University of Wisconsin-Madison. But it has its issues with reproducibility of cellular composition, and especially the structure of the tissues that are formed, and also the scalability, not only the long length scale in terms of culture, but also in terms of analyzing what's going on inside of these cultures. You have to basically look inside these millimeter-sized tissues, and that takes a significant amount of time. And so at Neurozetta, we've come up with our Rosetta RAID platform that we think alleviates a lot of these issues and standardizes the first approximately 6 weeks of human brain or spinal cord development, basically standardizing forebrain and spinal cord morphogenesis, both within cellular composition and structure of the tissue. So just to describe how we bioengineer human neural organelle morphogenesis. So we're again modeling the early stage formation of this neural tube structure that gives rise to all brain and spinal cord tissues. And if you take a transverse section, it basically is an annulus of neural stem cells, as indicated by the blue staining here, that polarize to the central lumen, which will become your ventricles and spinal canal, and deposit extracellular matrix proteins on the basal portion to wall off this CNS very early in development. If you take human pluripotent stem cells and you just differentiate them, whether in 2D or 3D formats, they spontaneously generate these neural stem cell populations, which form these polarized, what we call rosette structures. And they do this in a spontaneous manner in both space and time. And what we've done back in 2018 was to integrate this differentiation process with micropattern substrates so that we can now standardize the morphogenesis of these singularly polarized rosette tissues. And we array these on culture platforms within 96 well plates. And each tissue essentially is an aggregate of about 500 to 1,000 of these neural stem cells of the forebrain, of the spinal cord, depending on how the starting material that we have. And in the middle, you have standardized formation over the process of about a week, forming these polarized rings, these single rosette structures analogous of this annulus neural tube. And so what this has allowed us to do with considerable funding from NIEHS is to really standardize this as a screening platform. So we can take human pluripotent stem cells or neuromesodermal progenitors. The human pluripotent stem cells are used for forebrain. The neuromesodermal progenitors are used for spinal rosette arrays. We can validate and freeze down banks of those cells using protocols that we've developed over the last decade. We seed them onto micropattern well plates that we manufacture in-house. And then we can culture over the course of either 5 or 7 days, being exposed to environmental factors such as chemicals or drugs. And in a single 96-well plate, we can do full dose responses with 2 compounds per 96-well plate, while we also have our positive and negative controls. We actually, although there are 9,000 different microarrayed organoids on these plates, we actually only evaluate about 50 tissues per well. And so we're looking at 3,000 individual neural organoids that are being formed within this plate. And then we can run that through our AI-based Rosette analysis software called Rosette Detect. And this will analyze a plate within about an hour in order to spit out data on cell viability proliferation, the number of cells in each tissue, neurodifferentiation, which is the percent of those cells that are neural stem cells, and then rosette formation, actually analyzing whether you have formation and the wellness of formation of this polarized rosette structure. So a couple things to point out: this gives you really unprecedented statistical power to evaluate neural organoids with your compound and sort of the morphogenesis of brain and spinal cord tissues. This gives potential mechanistic insights depending on the juxtaposition of these curves as to which pathways that govern these different processes are being primarily affected by compounds. And really, this makes this entire thing really scalable. We're only limited by how many microscopes we have and how many people we have running these arrays. So this can be scaled to become a standard of practice. So we've done a lot of work that is now out on bioRxiv. This is a manuscript that's in review currently at a scientific journal, but we've put it on bioRxiv so you can see all the data. It's the full paper. And just as a sort of sneak peek, we have done a single concentration study when validating sort of the initial Rosette Array platform, where we've looked at a number of compounds, some that are standard developmental neurotox controls, as well as positive controls. And then we've also looked at a handful of pesticides, some that at the 10 micromolar concentration just kill our cells, so that's not really useful, but it was a single concentration study. But other ones that give more complex profiles for the different metrics that we pull off of the Rosette Array. We've also taken a handful of these pesticides and exposed them prior to putting them on the assay to pooled S9 liver fraction. So our assay can tolerate the human S9 pooled liver fraction, allowing us to simulate human metabolism before putting the compounds on our assay. And you can see from some of these compounds, the profile stays the same. Some of them, the profile changes where it becomes no longer a toxic mark. And other compounds such as ciproconazole, propizamide, thiocloprate, they become more toxic as you metabolize them. And just a note from sort of historical standard chlorpyrifos, we detect that both with and without human metabolism as a toxicant in our assay. That was recently restricted by the EPA. And so we have a number of compounds. We get really good sort of true positive, true negative sensitivity, specificity from this very limited study. So I want to say this is a limited study. We're doing a more comprehensive study that should be out hopefully before the end of the year. And most of these are right on target for true negatives and true positives. So we've, as I mentioned, are doing a much larger screen, and we have some initial results from that. And this is really just to indicate the type of data you can get back. So we have done compounds that are true negatives, and these would have no risk. We basically don't see dose responses for these compounds up to the 100 micromolar mark. If we have a dose response that is greater, I guess an IC50 of greater than 10 micromolar, we call that a low risk compound. Typically, these are products that are still in the market. And when you do IVE extrapolation, then they typically don't cause a concern. And then we have the medium risk compounds. These are depending upon the level of exposure. This will give you IC50s between 10 micromolar and 100 nanomolar. And really, this is reliant upon the proper in vitro and in vivo extrapolation to determine whether this is a concern. And then we also have the high-risk compounds. These are well-known developmental neurotoxins. They have clinical implications, and they typically give us an IC50 less than 100 nanomoles. And so if we screen compounds and they come in with an IC50 of less than 100 nanomoles, and that's probably something that should be looked at definitely further just with the results from the assay. So other things that we have done with this, I don't have time to go through all our results, but we know this works well with DMSO, so we can do both polar and non-polar compounds. We have really good reproducibility. This is all in the manuscript in more detail, where we get high efficiency for our control wells from forebrain as well as lumbar spinal rosette arrays. And we have a Z-factor score. Now we just keep on adding data to this as we do more assays of 0.53 across over 20 assays. We're able to integrate human metabolism if that's something that is wanted. If you know you have a compound that can be metabolized, then we can simulate that. We have seen that you get unique responses from the forebrain versus the spinal cord. And this is a particular interest because in most mouse models, neurotube defects show up as a forebrain defect, whereas in humans, the primary clinical occurrence is spina bifida, so in the lower spinal cord, and we can model both aspects. And we have done this not just for chemicals, but also for genetic factors that cause, for example, spina bifida. We've done 2 different genetic factors, and that data is in the preprint that I have at the bottom. And then we also have a Nature Neuroscience publication. This was a collaboration with Georgia Quadrato at the University of Southern California. We provided them our assay. They ran it with their cells that modeled autism spectrum disorder, and they were able to use our assay to get a really nice quantitative measure of the risk caused by a particular genetic background that's known clinically as a high risk factor for autism spectrum disorder. So I hope I've piqued your interest if you're interested in human developmental neurotox. And if you want to conduct these studies using our Rosetta arrays, Neurosetta is there and available for you. You can contact us at customer service or contact me personally. We have micropattern well plates and kits that we can provide. The plates we provide already and have done that to people, provide that to people around the globe. The kits we're beta testing, so please contact us if you're interested in beta testing a kit. Our Rosetta Tech software, the AI-based software, we are also beta testing, so please contact us there. And a lot of this work, I just want to acknowledge, was funded by a grant from NIHS, where they provided granting funds in order for us to scale up this assay to really take it through the ringer. And we're also doing a 100 compound screen with these funds. A lot of these compounds were provided by the EPA, so they also have supported this work. And this is a library that's an intersection of ToxCast and OECD DNT compounds. And with that, I can take any questions.
Thank you very much. I'll open it up to questions in the room. And I will ask one quick question. We've heard in previous discussions yesterday and today about the importance of representing genetic variability. And you had a little bit about that in your last slide about looking at genetic causes of neurodevelopmental adverse outcomes. How do you have sort of multiple donors whose genetics you've characterized, and what's the sort of impact on the outcomes that you see when you do tox screening, if you've done that across multiple donors?
We are actually trying to get funding to do that now. And so if there are people out there who want to fund that work, please contact us. I will be contacting people. And so, yes, we make these from human pluripotent stem cell lines. So we can do iPSC lines from all different backgrounds. We would love to explore that question in further detail. And so we're entering into the validation process now. So I'll be contacting you, Nicole, and others. And if we can get funding for that work, we would love to have a panel across various ethnic backgrounds and genders, because we have seen some differences with iPSC lines across genders. Across ethnic backgrounds, we're not sure yet, but we assume that would be there.
We have one question online. Emily, go ahead.
Yep. So for the speaker and others, where can we find the consensus list of agreed upon known positive and known negatives for use in vitro DNT assays?
That is an excellent question. And I'm a chemical engineer. And so I'm actually quite frustrated by the sort of endless search for the best list ever. What we have tried to hallmark or use as a milestone for our assay are things that are clinically known to cause human developmental neurotox. And so that's what we have really tried to look at, things that are known to cause, for example, neurotube defects. There is no really good, well-known cause for autism, although there was a nice New England Journal of Medicine paper that came out. And so we're actually testing those compounds that they showed. And so typically we look at the OECD guidance. There's also Altex had a list that came out a couple of years ago. So those are some good references. I'm happy to provide those to you. But for us, we really benchmark our assay based on clinically known compounds or genetic variants that are known to be well associated with developmental neurotox.
Thanks, Randy. And yeah, I would encourage you to put the links to those references in the chat. And yeah, I'll just reemphasize the OECD guidance document that was just endorsed and published last year. Another just piece of information is that there is an international consortium, and this specific task is being led by Kevin Crofton and Bill Mundy to update the reference list and include in vivo information and human information. And that list will be published very soon on EFSA's website. And once that's available, it will also be integrated into ICE. So we have chemical lists, as most people are aware, in the integrated chemical environment. So we'll make it available through that as well. Okay, thank you very much. We're gonna have to move on. So our next presenter is Maricel Marin-Cuan from Nestle. And let's see, Maricel.
Yes, hello.
Okay, so you are sharing your slides. So
Yes, can you hear me?
Yes, we can.
Okay, great. Is it okay, the sound?
Yes, if you want to turn your video on, you're welcome to. Otherwise, go ahead.
Okay, great. So hello, everybody. I am representing AUSC Europe section with an initiative on best practices for bioassays testing of food and other complex mixtures. And yes, this is a very interesting public forum to share this kind of information, because the overview that we want to share today is that we know that there is growing attention to the use of in vitro methods to identify potential hazards of concern and understand mechanism of toxic effects with relevance in the human context. However, we know that to find this alignment for the transition to animal feed regulatory system for safety assessment of chemicals and mixtures, it's really challenging, mostly for the food, packaging, or environmental context. And we strongly believe that we need a framework or guidelines towards the validation and harmonization of these bioassays methods in order to really build a robust ground and reliable data analysis and therefore biological interpretation. And I think that's why we are very happy that there is taking place this public forum, because yesterday the discussions are really going into this direction. So this is very positive. And which is important and impact in the field that we want to share today is that the suitability of the in vitro methods are typically validated for their use with biochemicals, but for example, for complex mixtures, including food, environmental sample, or packaging, or any other mixture, is not necessarily the case. And there are many underlying issues in the in vitro field for applications that we really need to improve best practices, for example, mixture preparation, accuracy, reliability, reproducibility, extrapolation. Data interpretation is a very, very important topic. We have an experience that we did an exercise sending one sample to 5 different CROs to see which is the decision or in vitro data that we get back. And actually we got 5 different decisions about the biological activity. So we said we really need to align this topic, because it's very important for many experts and stakeholders from packaging, environmental, like water, soil, agro industry, but also food and beverages, and also people working on raw material suppliers and technology providers. This is a very important topic. And this is very important now because we are in this transition for the harmonization and acceptance of in vitro practices by regulatory authorities. That's why we are talking today, to go to this 3R alignment. And Of course, we know, and I think we are aligned with all the discussions from yesterday and today, that there are critical limiting factors for reliable approach to characterize complex mixtures. And the challenges and gaps are really very, very important because we really need to build a robust ground considering drivers and limitations when defining the requirements for qualification, how to get reliable data, and how to analyze this data. And of course, the validation of the bioassays for complex mixtures, and to try to harmonize in order to really facilitate the acceptance is by regulatory authorities is really critical. Some of the identified gaps that we have pointed out is the integration of fundamental upstream boundaries, including, for example, end-to-end processes. We are talking a lot about new technologies, sophisticated methodologies, but we are forgetting how to prepare the sample. If you prepare the sample differently than another laboratory, we will get another response. So this is integration of really end-to-end that is very important, not only the development of new methodologies or alternative methodologies, but also the sample preparation to know what are we preparing and at what we are getting with the bioassays in order to align with the data interpretation and the extrapolation guidance. So the status of this activity that was initiated by AUAC Europe, like in 2022, we organized a workshop and in Amsterdam in 2023. And we gathered some people that were concerned about this topic, and we created working groups on key identified topics on food and other mixtures, like what we are addressing in this conversation is the extraction methodologies, how to consider the type of mixtures or to work, for example, on categories in order to really to align when we address mixtures, what are we really measuring. Another working group is working on the framework for qualification and data interpretation, and really defining modeling and criteria to analyze mixtures, because the extrapolation from in vitro to in vivo in the case of mixtures is very challenging. And the third group is really trying to align with regulatory landscape to enable and driving the acceptance for mixtures. This is very important, as we are discussing yesterday. You know that the flexibility of regulatory and also this training and understanding that alternative methods are probably necessary in order to address the development of NAMs. And at the end of the day, what this working group want to achieve is to generate a state-of-the-art review publication on best practices for mixtures, including a group opinion to be published in 24-25. And the working groups will identify drivers in the area for future validations, like, for example, mixtures categories, for example, sample preparation and extraction, in vitro qualification, and to consider really the importance of the harmonization for the data interpretation in order to really fulfill the acceptance of the NAMs in the application for mixtures. We will have coming project meetings, AUAC International, that will take place in August in Baltimore, and at the end of the year, all the working group members we will meet in Prague for the RAFA meeting. So you are all welcome, and if you have questions, please don't hesitate to contact us. And if there is interest in this working group, you can contact the AUAC Europe President, Thomas Good, that will be happy to give more information. And that's all what I wanted to share today, and open if you have questions.
Thank you
Sorry,
very
I
much.
forgot to turn on the camera.
Nice to see you. Thank you very much, Maricel. And I will just note that I hope that you're able to stay for the rest of the day, even though it's a bit late probably where you are.
Yes.
that after lunch, we will have an excellent presentation from Cynthia Ryder from NIEHS, who's going to talk about the Botanical Safety Consortium and how they've been evaluating NAMs for use with complex mixtures.
Exactly. Yeah. Yeah, I saw that in the program. I hope it's not too late, but I will try.
Well, we're doing well at staying on time so far. Any questions in the room? Online? Nope. Okay. Thank you very
Okay,
much.
thank you.
So now we will go to our next presenter, who is Sherman McFarland from the Johns Hopkins University School of Public Health. Sherman, will you be sharing any slides, or are you just speaking to us today?
No slides, just speaking.
Okay, great.
Is my volume okay?
The volume is fine, and let me just pin your video so folks in the room can see you. Okay, please go ahead.
Thank you very much. Dear ICVAM, members of the committee, member agencies, and esteemed guests, my name is Sherman McFarland. I'm a postdoctoral fellow at the Johns Hopkins University School of Public Health. I work for the Johns Hopkins Toxicology Policy Research Group, which is comprised of scientists and lawyers. And we appreciate the ability to submit written and oral statements at this forum. The goal of the Johns Hopkins Toxicology Policy Research Group is to advocate for the development and use of new approach methodologies, or NAMs, and increase their use in regulatory decision-making. I should note that I am making these statements today in my individual capacity, and that the views expressed do not necessarily reflect the official policy or position of Johns Hopkins University or the Johns Hopkins University Bloomberg School of Public Health. Now, the purpose of this statement is to make 4 recommendations to ICVAM. First, promote data sharing and stakeholder involvement. ICVAM should provide guidance and resources for data owners to share data sets in compliance with confidentiality regulations. Data sharing and accessibility play a crucial role with regard to increasing the use of NAMs. Facilitating data sharing among researchers, regulatory agencies, and other stakeholders enables collaboration and enhances the reproducibility and refinement of NAMs. Second, collaboration and harmonization with international partners with respect to validation and approval. It is imperative that ICVAM and its member agencies collaborate with international partners such as the European Union and OECD to validate and approve NAMs and harmonize their acceptance worldwide. We believe that working with international partners will enable agencies to validate and accept NAMs at a quicker pace. We were glad to learn about collaborations with OECD and urge ICVAM to find other international organizations with which to work. Third, establish timelines and goals for ending animal testing. ICVAM should encourage federal agencies to reduce animal testing as rapidly as feasible. Setting such goals is important, and we suggest that agencies could and should reduce animal use by 50% in 5 years and end all animal toxicology testing within 15 years. ICVAM should advocate for these goals and assist agencies in reaching them. ICVAM should also encourage agencies to annually report on the progress that they have made toward reaching these goals and identify any and all successes, failures, or difficulties the agencies have encountered with respect to reaching these goals within the agency's annual reports. We were encouraged to see that EPA is moving to report the number of animals used, and we believe that other agencies should follow suit. Fourth and finally, build scientific confidence. ICVAM needs to take a more proactive role to ensure that NAMs are validated and accepted by regulatory agencies within realistic timeframes given resource constraints. ICVAM must also emphasize to policymakers, scientists, and the public that the goal of NAMs is to produce scientifically reliable results. Such an emphasis is critical for garnering support, recognition, and acceptance of NAMs. By taking these 4 steps, ICVAM can contribute substantially to efficient, ethical, and scientifically sound approaches to toxicity testing and advance animal welfare, public health, and environmental protection. Thank you very much for the opportunity to speak today.
Thank you, Sherman. Are there any questions in the room? Nope. Any questions online? Okay, Thank you. Appreciate the clear presentation. So we will now go to Vicky Kachernak, who I believe is also online.
I am, thank you.
And Vicky, do you have slides to share, or will you be giving us an oral comment?
Just an oral comment, thanks.
Great, hold on, let me just pin your video so everybody can see you in the room. Okay, please go ahead.
Thanks, Nicole. My name is Vicky Katrinik, and I'm the Director of Animal Research and Testing at the Humane Society of the United States. Thank you for the opportunity to comment today on behalf of HSUS and the Humane Society Legislative Fund. We have submitted more substantial written comments, so I'm going to try to be really brief today. I always appreciate the ICVAM Public Forum and the chance to hear about all the great work happening at all the agencies. But there are a few things we'd like to highlight. We believe it's important that any work to build confidence in NAMs also includes recognition of limitations of animal data. Without a proper understanding of the inherent problems with variability and uncertainty of data from animal tests, NAMs will seemingly be held to a higher standard. ICVAM member agencies should conduct retrospective analyses on animal data that have been submitted to them and specifically examine what portion of those data was actually used in regulatory decision-making. As just one recent example, in 2023, a retrospective analysis was published that looked at the need for the 90-day dog study for pesticides. The result of this analysis was that for most pesticide risk determinations, the dog study provided no benefit beyond data that was already submitted for other purposes. This example demonstrates the importance of critically evaluating the continued regulatory need for certain animal data. In order to ensure that efficient transition from animal tests to NAMs, all ICVAM agencies must clearly communicate with stakeholders about the acceptance of NAMs, ways to eliminate unnecessary animal testing, and appropriate processes to ensure consideration of new technologies. ICVAM's public forum is a unique opportunity for member agencies to share the work they're doing, figure out ways to work collaboratively, and communicate with stakeholders about what still needs to be done to build confidence in new approaches. Discussions of ways to avoid unnecessary animal testing and incorporate NAMs into testing plans should become a regular part of agency interactions with regulated industry. We also encourage ICVAM member agencies to consider opportunities for incentivizing NAMs use and data submission. To ensure acceptance of data from NAMs, it's important that developers are granted an opportunity to present their new technologies to agencies. For example, FDA's iSTAND pilot program. The 2022 guidance document from the Consumer Product Safety Commission informed stakeholders on the process by which staff assesses whether alternative approaches are appropriate for use. We encourage agencies to devote additional funding and staff time to support ensuring the success of these types of programs. A barrier to the replacement of animal tests is the lack of understanding about what data from animal tests are or are not required, and clarity about when NAMs can be used instead. Guidance documents should be regularly updated to reflect the most recent science, and outdated guidance should be removed to avoid confusion. As NAMs become accepted for specific tests, guidance documents that encourage animal use for those tests should be promptly updated to avoid any unnecessary animal testing. We also encourage ICVAM member agencies to release or update strategic plans to reduce animal use, create timelines for progress, produce metrics for tracking uptake of NAMs, and provide the incentive needed to ensure NAMs are fully incorporated into regulatory decision-making. It is also important that ICVAM member agencies work to increase international harmonization, maintain consistent communication with regulatory agencies outside of the US, and continue to participate in international organizations. The final point I want to make is about the importance of prioritizing funding for NAMs on the agency level. We were pleased to see the creation of NIH Common Fund's complementary program. We appreciate this forward-thinking approach to addressing challenges in human health while also investing in development and ultimately regulatory acceptance of NAMs. In fiscal years 2023 and 24, Congress included significant new funding for FDA to reduce animal testing through alternative methods. We encourage agencies to advocate for dedicated funding for NAMs and we will continue to urge Congress to provide appropriations for the prioritization of these agency efforts. HSUS and HSLF welcome the opportunity to work with NICEDM or any ICVAM agency to help achieve the common goal of replacing animal tests with human relevant methods. Thank you so much for your time.
Great. Thank you very much, Vicki. Are there any questions in the room? Nope, online? Nope, okay. Thank you. And we will now move to our next public commenter who is here in person. So let me just reorient. Do you have slides? No slides. Okay. So Shagan Krishna from the Physicians Committee for Responsible Medicine will just give us an oral comment. Here you go.
Hello, everyone. Can you hear me properly? Okay. My name is Shagun Krishna, and I recently joined the Physician Committee for Responsible Medicine as a computational and in vitro toxicologist. And while I'm new to the team, so my comment today reflect the collective thoughts of many dedicated PCRM members, including Elizabeth Baker, Dr. Erin Slangster Schmirer, Dr. Catherine Krabs, Joe Manipule, and Emily Anderson. And we all deeply appreciate the ongoing collaboration with NICEATM and ICVAM and look forward to continuing our work together. And first, we want to thank NICEATM and ICVAM for their leadership and dedicated work over the past year. There has been significant progress in advancing human-based science, which refocuses research and testing on human biology while also reducing the use of animal methods. This is a vital shift in our approach to toxicology and medical research. As we continue to advance in this field, it is crucial to recognize the importance of supportive policies in sustaining scientific progresses. To support these scientific advancement, we urge agencies to review and update policies, incorporating feedback from stakeholders on how regulation and guidance should evolve to support the use of these NAM-based approaches. We believe that this kind of responsive and adaptive policymaking is essential for the broader adoption for the NAM-based approaches. And we are very encouraged by the increasing availability of the training programs. We have seen great strides with programs like EPA, ASCCT, NEURA, and EPIC. And we thank everyone who was involved and encourage agencies to communicate any specific training needs so that these programs can be tailored to meet them effectively. And one thing we would like to mention that it would also be beneficial if these programs should be broadcasted widely to reach a larger audience across the world, because we feel that everyone should adopt these methodologies. And additionally, we believe that more training programs should be conducted on how to validate these NAM-based approaches. And we are also pleased to see significant momentum at the NIH over the past year, from the adoption of ACD working group's recommendation to the launch of complementary program, which I believe is a milestone. These efforts are foundational in refocusing health and disease research on human models, and the strategic planning for complementary has been particularly exemplary, where they have involved the broad stakeholder input and the public engagement. We at PCRM applaud these inclusive approaches and encourage ICVAM representatives to share these successful strategies within their respective organization. So with this, I would like to conclude my comments. We have already submitted a comprehensive written comments. And thank you once again for opportunity to speak up at this forum and for your continued commitment to advancing human-based science and reducing animal testing.
Thank you, Shagan. Any questions in the room? Online? No? Okay. Moving right along, our last but not least public commenter is Christy Sullivan from the Institute for In Vitro Sciences. Christy is online, and Christy, I have your slides. Just give me a second to pull them up.
Okay, thanks, Nicole.
Now we should be good.
There
Okay,
we are.
go ahead, Christy.
Okay. Oh, thanks. Thanks very much. Well, I'd like to say thanks, first of all, to Nicetum for squeezing me in. I, like others, have really appreciated the discussion and the dialogue so far at the forum. I want to talk a little bit about education and outreach. These aren't directed at any particular agency, but an invitation to learn and collaborate to all ICFAM member agencies, to Nicetum, and to other stakeholders. Can you go to the next slide, please? So my name is Christy Sullivan. My title is Vice President for Outreach and Education with the Institute for In Vitro Sciences. Many of you are aware that we are a GLP in vitro testing laboratory right up the road in Gaithersburg, but as my title implies, we do a lot of work in education and outreach. And so from our perspective, what that looks like is maybe a little bit broader than what you may have thought previously. I'm sure you think, of course, about the traditional educational activities like webinars, in-person trainings, but our work also includes a lot of validation of methods and approaches, and evaluation and implementation, and then communicating that information and our experience through outreach to agencies, engagement with the regulated community, and helping to set international standards. And importantly, all of this activity is very iterative. So it really, we think it's important to make sure that as we gain more experience, we continue to integrate that into our training and education. Next slide, please. So I'm really looking forward to the publication that Dr. Harrell mentioned yesterday, outlining some of EPA's experience with next generation risk assessment approaches. There's clearly a lot of activity going on in developing NGRA, and we are in the middle of a transition point in applying these approaches for regulatory use. But to see what, as you can see on this image, which I took from Gavin Maxwell, he's nicely depicting here as going from part of the community, really developing and demonstrating through case studies about how to use these approaches, to that shift where the majority of the field is applying them for fully non-animal risk assessment. Education and outreach is the key to seeing that shift. We think over my career, I have learned that this is one of the things that addresses many of those barriers to acceptance that we can't quite necessarily put our finger on scientifically, right? They're about comfort and experience and getting familiarity with new approaches. With an educational program, we can show how methods can be applied, tailored to the audience's specific needs. I think in toxicology, the field is very unique in that we all have very variable education and career pathways that have brought us all, even in this room together, but into the field together. These programs help us level that field and make sure that we're all in the same place. And obviously, I think both from the industry perspective, it increases the likelihood that those methods will be conducted and used in their safety assessments, but also used by regulatory agencies to make decisions. Really importantly as well, it makes sure that when people are conducting in vitro methods or computational methods, that they are doing them in a way that is consistent and of the best science, so that we can ensure that these the science is trusted. Next slide, please. So over the past year, we've done a lot of horizon scanning and a number of trainings. We do about anywhere from 5 to 10 in-person laboratory hands-on trainings per year, both in Gaithersburg and around the world. And some of our priorities that we've got in line for the community to focus on are on the slide here. I want to in particular focus on data analysis and weight of evidence. We see that a lot of people know how to, or at least are relatively familiar with a lot of the in vitro methods, but where the opportunity for discussion and engagement is about analyzing that data, analyzing what happens in the laboratory and how to make sure that that is quality data coming out of the laboratory, to kind of choosing information sources that get you to the answer you need, as well as combining all of the data into a weight of evidence to make a decision. That is a lot of where we're putting the focus through case studies and examples that we've experienced or other people have. As well, we are going to be really focusing on good in vitro methods practices and ramping up our activities on training the trainer. There are a lot of training activities going on, but we need to increase the capacity both in the US and internationally. And so the next step, I think, is making sure that training courses can continue beyond the folks that do it now, or sort of the key agencies that are doing it now, like EPA. And then, we're talking a lot about gaining confidence. And so let me go to the next slide. And, reflecting on confidence building frameworks, whether one published by Van et al. On the slide here, or the one that ICBAM just published in its validation report, we can identify opportunities for sharing experience and lessons learned on how to characterize these methods or approaches in alignment with those frameworks. And so I'm thinking here on the sort of the right side of the diagram on independent review and data integrity and transparency, and even technical characterization, proceeding through those aspects of the framework, or even reflecting effectively and transparently the information about a particular approach from the test method developer perspective. There is an opportunity for education and training to help make sure that those new approaches are reflected well. And possibly the method developer forum is another opportunity for that. Next slide, please. I wanted to point out that there is a lot of guidance now on good in vitro methods practices from the OECD, from our own organization, and then we also collaborated with the EU EPPLAS training platform module to develop that, and there should be more as well coming online. And next slide, please. So there are a couple of in-person trainings coming up. First of all, tomorrow, there's one on the Nicetum ice, which is a hands-on training. Be sure if you're going to be in town to take advantage of that. The QR code is, we're just announcing, that's a link to our Tox Tracker training, which is going to be in August, focused specifically on the Tox Tracker genetic toxicity assay. And then we have a couple of spots left for the in-person practical methods laboratory training in September. And I think that's the end of my talk. I got one more slide, and while I'm there, I will just go ahead and take a picture of these links or something, because there are so many resources available, whether you want to learn about NGRA, or go to a webinar, or dig into the EPA CompTox dashboard. There's a lot of opportunity to get engaged. Thank you very much.
Thank you, Christy. I'll buy you that drink next time I see you for highlighting the ICE training. All seriousness, for those of you in the room, or those of you that have colleagues locally, hopefully you are all aware, and many of you are already registered, but we do have a hands-on integrated chemical environment training tomorrow afternoon here in Bethesda, here in this building. I have a quick question for you, Christy. One thing that I think IIBS has been pretty engaged in, that I was wondering if you could expand on a little bit more, is international outreach and training. And I'm thinking in particular about regions of the world that may be, socioeconomically depressed or not have access to the level of resources that it takes to develop or build NAMs themselves, but are also simultaneously disproportionately suffering from environmental chemical exposures and really need access to more rapid, more efficient resources to be able to make decisions that can better protect public health in those regions. Can you comment on maybe opportunities to increase our outreach into those regions of the world or areas in which we need more focus?
Yeah, I think for me, one of the key opportunities is, or maybe the key first steps is there is always someone in each country who is kind of has raised their hand as someone who's interested in vitro or non-animal approaches, whether it be a laboratory or a university professor, or in the case of Brazil, there are organizations like BRACFAM. And that's the place to start because they tend to be sort of the national connection point for figuring out how to engage with regulatory agencies and other key companies, for example. To me, I think it's a combination of sharing the opportunities for the computational tools and really kind of walking through in a hands-on way, here's how you can use some of these to get at least some information, and then following that up with, I think some of us have a lot of experience with setting the foundation and growing in vitro resources step by step, and can share a lot of those experiences with labs as they grow in different countries. And of course, what we find is that the entire stakeholder community is really helpful in providing resources in that way. So you have companies, NGOs who are interested in helping to provide equipment, and things like that will help increase that capacity.
Okay, thank you. Any other questions or comments in the room or online? Okay. Thank you very much. And so that concludes our morning session. I want to thank again each and every public commenter that provided both written comments and oral public comments. They were extremely thoughtful. I can tell a lot of time and energy was put into providing us with really valuable feedback that will continue to guide our work moving forward. So I just want to express my deep appreciation for all of our public commenters. So we will now take a 25-minute break and we will reconvene at 11:15. I will quickly whip up a slide to indicate that for our folks online. And for everybody in the room, we'll see you back here at 11:15 sharp. We have a very exciting next session, special session from our NIH OD colleagues and our FNIH colleagues to discuss complementary and activities of the ACD NAMS working group and everything in that space. So see you in 25 minutes. Thank you again. Wait, no, 11:15. 11:15, that's in 15 minutes. I actually have a degree in math. 15 minutes, see you at 11:15. Okay, so thanks everybody for keeping to time on our 15-minute break. So I am super excited about the next session. Those of you who know me know that I tend to be excitable, but this session in particular, I think is really remarkable because it highlights sort of the intersection between biomedical research and the work that we've been doing in ICVAM and NICEDM to advance NAMs for safety testing, and sort of the importance of investment and advancement along every piece of that continuum, and how critical it is for us to maintain communication with our friends and colleagues doing basic science and mechanistic research, and put resources into validating and qualifying those methods so that they can then be used in regulatory decision-making and in regulated industry. So that's my little intro spiel. But it is my great pleasure to introduce Anna Masuko, Dr. Anna Masuko, who is a senior science advisor in the Office of the NIH Deputy Director. And she is going to be speaking to us today about the recommendations from the NIH Advisory Committee to the Director Working Group on NAMs. And let me go ahead and share the screen. Anna, the floor is yours.
All right, thank you very much, Nicole, for that kind introduction. And I'm really excited to present today and just give you sort of an overview perspective from the NIH on how we're thinking about NAMs overall, and specifically as it relates to the NIH Advisory Committee to the Director, which recently had a working group specifically focused on novel alternative methods. So I hope this will be kind of relevant to all of you as sort of a big picture context coming from the NIH. So with that, on behalf of the NIH Office of the Director, I'm representing the Division of Program Coordination, Planning, and Strategic Initiatives within the NIH Office of the Director. So just to take a step back for a minute before I kind of dive more into the advisory committee to the director and their recommendations focused on NAMs, I just wanted to show a quick snapshot really of the last 40 years of NIH investment in this space. And what I wanted to just highlight for all of you is that even though this working group was launched and given its recommendations only in the last year and a half, that overall there's been a significant increase in activity in this space and NIH's support for it, as you can see, pretty significant even around the early 2000s. So this is sort of the context of just increase activity in this space. And I think for this reason, the NIH and the NIH director felt that it was really timely to better understand the state of the science and how we can really leverage all of this ongoing activity in a more coordinated way and really kind of set a future direction for this area of science overall in as kind of strategic and coordinated a fashion as we can. So with that as a little bit of context, the NIH director charged a working group in 2022, and it first met last January. And the charge of this working group was to identify the types of alternative methods and assess their current strengths and weaknesses for studying human biology, circuits, systems, and disease states, to characterize the types of research, condition, or disease for which NAMs are most applicable or beneficial, and articulate high priority areas for NIH investment in the use and development of NAMs with human applicability to advance progress in understanding specific biological processes or states, and to augment the tools and capabilities for biomedical research to complement and or potentially replace traditional methods. And so here you see the members and co-chairs of this working group, which was established with diverse perspectives across this area of biology from academic institutions, government partners, and industry to really look at this problem in a holistic way and provide recommendations to the NIH director for how we can better address this area. So before you is just kind of a snapshot of the timeline of the working group, its major activities, And what I'd like to just point out, which I think is really significant, you can see summer 2023, public input was sought through a request for information. Also, a public workshop was held last year. And so it was really important for this group to garner input kind of from across the landscape to really inform its recommendations. And last December 2023, the recommendations were presented and accepted by the advisory committee to the director. And in February of this year, our new director, Dr. Bertignoli, accepted the recommendations and has charged us to move forward with their implementation, which is what I'd like to talk to you a little bit more about today. But first, just to highlight kind of what was heard from the community. Overall, the themes from the RFI and the public engagement was really the importance of integration. Clearly, there are high priority needs. And then you can see several other themes on this slide. And what really came through was the importance of integrating across these different areas, especially involving combinatorial NAMs, interoperable data sets, technology dissemination, coordinated infrastructure, multidisciplinary teams, comprehensive training, and socially responsible technologies. So these were the major themes, and I think throughout them, you can see how integration across several levels, both as far as the technology itself, but also the workforce overall and the community that will be developing and implementing NAMs, that integration was really a key theme. So here are the recommendations themselves. There were 7 key recommendations. There were also several sub-recommendations underneath each of these recommendations. So I won't go into the details on all of the sub-recommendations, but I'll quickly take you through the major 7. And then I will say also that the full report of the advisory committee working group is also on the ACD website, on the NIH website, if you'd like to download the full report. But in summary, here are the 7 recommendations to catalyze the development and use of NAMs. First, to prioritize the development and use of combinatorial NAMs. Second, to establish resources, infrastructure, and collaborations to promote the use of interoperable, reliable, and well-curated data sets produced from research using NAMs. Promote effective dissemination and interconnection of NAMs technologies, invest in comprehensive training to bolster continuous advances in NAMs development and use, facilitate multidisciplinary teams with expertise across technologies and the life cycle of NAMs development and use, promote social responsibility in both the creation and deployment of NAMs across the research life cycle, and support and maintain coordinated infrastructure to catalyze effective and responsible NAMs development and use. So I'm just going to go into a little bit of kind of unpacking these recommendations, just a few slides, and then I'm going to end with how we're viewing the implementation of these recommendations and just give a highlight or two of ongoing work at NIH, which you'll hear more about in the next presentation. So recommendation 1, prioritize the development and use of combinatorial NAMs. This is really focused on technology combinatorial effect, a phenomenon where the integration or combination of different technologies or their components results in a more significant impact than the sum of their individual effects. And it's really focused on strategic combination of NAMs that can lead to breakthroughs not possible with any single approach. Secondly, focusing on reliable and interoperable high-quality data sets are needed for appropriate validation or qualification of NAMs. Appreciation for the fact that data and associated metadata should be generated both from NAMs and traditional approaches, and that high-quality data increases overall efficiency, reproducibility, and validity of comparisons. Thirdly, that effective and rapid technology dissemination really requires NAMs developers to consider users through the development process. That successful deployment requires clarity regarding technology maturity for use and dissemination, which I think is particularly relevant for today's meeting, and the importance of designing to be fit for purpose. Fourth, that technology dissemination is necessary but not sufficient. It needs to be combined with support for the skills and knowledge to use NAMs appropriately, with a particular focus on ensuring equity, both in terms of institution, career stage, and role in the development process. And that a key to success is integrated and interdisciplinary collaboration. There was also a focus on collaboration being needed across multiple sectors, disciplines, and expertise across the life cycle of development. An acknowledgement that incentives are lacking for integrating cross-disciplinary research with implementation in mind, and a recognition that there are currently different lexicons and approaches which create barriers to communication across fields and sectors. There's also a really significant recognition of the role of social responsibility in this process, including the fact that trust throughout the life cycle of NAM's development and use is required to promote its uptake in the community, a focus on ethical considerations upfront to ensure inequities are addressed, both in terms of design and ultimate access and cost, and an acknowledgement that while the opportunity is clear, we need to be sensitive to ensuring that narratives don't overly promise on what NAMs currently can deliver, and that we really need to have authentic communication about where the science currently stands in order to ensure that NAMs are ultimately successful and well implemented. There is also a clear need for dedicated resources and venues to support NAMs. Venues for communication and collaboration include creating organic environments for common language and standards, reducing redundancy to maximize our investments, and spurring new collaborations for integrated approaches. So as you can see, I hope from these 7 major recommendations that the vision here is really an integrated ecosystem to catalyze scientific discovery that focuses on development of combinatorial NAMs, interoperable, reliable data sets, and effective technology dissemination. And that to do all of those things, we really need a community where there is comprehensive training, multidisciplinary teams, social responsibility, and coordinated infrastructure throughout the entire process. So where are we now? As our NIH director accepted these recommendations in February, and we are now moving forward towards implementing this vision. So our division within the Office of the Director has been charged with coordinating the implementation of these recommendations across NIH. So as a first step, we've conducted a prioritization and a feasibility analysis of all the recommendations and the sub-recommendations, and we've identified lead institutes and centers for each of these recommendations in order to really understand kind of who's on first across the whole agency, which offices and institutes have the appropriate expertise to carry these recommendations forward. We've held preliminary meetings with the most heavily involved offices and programs, including with Nicole and ICVAM. And where we are now is focusing on developing metrics for success and processes for tracking our progress in implementing these recommendations. And we hope to soon engage all of the NIH institutes and centers, as there were several recommendations which all NIH institutes have a part of their scientific portfolio that's relevant, so that we can really make sure we have a clear vision and really understand all of the activities across NIH that are relevant to these recommendations, and ensure that we can track our progress and make sure that this vision can go forward. And ultimately, we plan to provide an update to the advisory committee meeting, their next meeting in December of this year, to the ACD and the NIH director on what we've done to date to implement these recommendations. So I want to just leave you with, to set the table, I hope, for the next presentation from my NIH colleague, is just a quick snapshot on one program that's already underway, which we're really excited about because it aligns so well with several of the recommendations from the ACD working group and is already underway. So the Complement Animal Research and Experimentation Program is focused on catalyzing the development, standardization, validation, and use of human-based new approach methodologies, or NAMs, that will transform the way we do basic translational and clinical sciences. This is being launched from the NIH Office of the Director within the Common Fund. And this program was approved in January 2024 and is already underway. And you'll hear more details, but just several kind of sneak peeks at some of its activities. Complementary has already held 3 listening sessions with major collaborators on this work. A federal interagency retreat was held here at NIH last fall. And just several weeks ago, prize awards were announced for an ideation challenge that was supported through this program. And it is currently having further conversations to explore public-private partnerships to address several of the other goals of the program, which you'll hear more about in the next presentation. So with that, I'm happy to turn it over to my NIH colleague from the NIH Common Fund, or take any questions if we're holding to the end.
Thank you so much, Anna. Do we have any questions? Yes, go ahead, Paul.
Thank you for that really interesting presentation. My name's Paul Locke. I'm from Johns Hopkins Bloomberg School of Public Health. I have just a specific question for you about the slide where you showed the increase in, I guess, NAMS-based research. We have tried to find that information, and we have found it impossible using the public tools that are available to us. So I would urge you to make that data publicly available, and also, to the extent you can, to provide us with the underlying data that allowed you to build that graph.
Sure. As far as I'm aware, it should be directly from the ACD working group report, which we can certainly send to you. And there should be details of how they did that analysis, but happy to follow up and make sure you get the information. Absolutely.
Thank you.
Mhm.
All right, we have one question that was related to where the working group report is housed. I'm putting that link up into the chat for you.
Okay.
And then another question is, will a formal implementation plan be published based on the recommendations?
Yeah, that's a great question. I don't think we've made a final determination on, the need for a formal implementation plan. We certainly have an internal implementation plan that's in development. Some of the discussion related to prioritization, feasibility, and identifying the key lead offices. And we do have kind of an overall timeline for implementation, and where we are now is finalizing the metrics to be able to track our progress. But I think, development of a more formal implementation plan is certainly something kind of still in discussion.
Thank you so much, Anna. And now I would like to welcome Dr. Margaret Ochinska, who is going to give us many more details about the complementary program. Let me just pull up the slides and.
Good afternoon, everyone. Thank you very much for the opportunity to present an update on the complementary program, and thank you for that great lead up to my presentation. I'm very happy to provide this update and give you a bit more context about what we have been up to at the NIH and the Common Fund to address implementation adoption of new approach methodologies. So my name is Margaret Ochinska, and I am the program leader or program director of the Reengineering the Research Enterprise Division within the Office of Strategic Coordination in the Common Fund. And I will talk to you today about the complementary Complement Animal Research and Experimentation program that has recently been cleared for implementation. So I just wanted to give a little bit of a historical context. So in terms of where the Common Fund comes from. So this is sort of going back to the historical aspects that we heard earlier this morning. Where do we come from? What are we doing? Where are we going? So essentially in 2004, the Common Fund initially was developed as an NIH roadmap where institutes and centers would contribute funds to develop trans-NIH collaborative projects. But in 2006, when the Congress unanimously reauthorized the NIH, they actually created the new Division of Program Coordination, Planning and Strategic Initiative, DPCIPC, that Anna Mizuka was from, and within the Office of the Director. And the NIH Common Fund was authorized and received its own budget. So it no longer takes contributions from institutes and centers. It has its own set-aside budget similar to an IC. And so in terms of the Common Fund, the purpose of the Common Fund is to support bold scientific progress, programs, and catalyze discovery across all medical and behavioral research. And it advances areas of biomedical and behavioral research important to the mission of multiple institutes and centers. So that's the key. It's not just the mission of one institute and centers, it's multiple ones. And it's supposed to spur subsequent biomedical advances. And so how does it do it? It makes substantial investments in time-limited, goal-driven programs, such as complementary to significantly change the trajectory of biomedical research by accelerating emerging science, removing research roadblocks, enhancing the research workforce, and supporting high-risk, high-reward research. And so in a sense, as the Common Fund concept is developed, it engages scientists from diverse disciplines to provide input in program planning. So the strategic planning efforts that were described involved input from multiple stakeholders across the landscape. And we also invite researchers to participate in interdisciplinary consortia to tackle shared goals. And it includes leadership from staff from institutes and centers and office director that work together to design this implementation to oversee the projects. And so really the complementary program catalyzes the development, as was mentioned, standardization, validation, and use of human-based new approach methodologies. So that was a concerted effort to focus on human-based methodologies. So human-derived, human-based data derived from human subjects that will transform the way we do basic translational and clinical sciences. The goals are to better model and understand human health and disease outcomes across diverse populations, to develop NAMs that provide insight into specific biological processes or disease states, to validate mature NAMs to support regulatory use and standardization, and to complement traditional models and make biomedical research more efficient and effective. So that was also a very thoughtful way to create the name of the complementary program, because really it is to complement existing systems, but to enable more utility and adoption of new approach methodologies. So I wanted to also recognize this concerted effort across all the institutes and centers at the NIH to help make this possible. So we have about 23 institutes and centers and office representatives on the working group. And the effort was spearheaded by NCATS and NIEHS, by Joni Rutter from NCATS and Rick Wojcik from NIEHS. And we have working group coordinators, Dan Tagley from NCATS, and Dan Shaughnessy from NIEHS. And then we have the strategic visionary in the room and with us as we develop the strategic planning effort, Nicole Kleinsteuer. And then we have additional key members of our core team, Katie Melora and Tony Casco from the Common Fund. And then we have multiple working group members across the institutes and centers, as I mentioned. So we would like to thank for their contributions into developing the strategy. So essentially, just to give you a bit of a timeline for how this occurred, you've heard from my colleague, Dr. Mizuko, about the ACD working group on catalyzing the development and use of NAMs. So that activity began last year as well, and there was a presentation of the preliminary findings. While that was ongoing, our complementary working group, the concept was cleared to start with the strategic planning phase. And so that was back in June. The concept proposal and the complementary working group was convened. And so the activities that ensued after that working group launch included a deep landscape analysis, listening sessions and roundtables, an interagency workshop, and an ideation design prize challenge that was used to inform as a crowdsourcing method to understand what is out there in the community, what ideas arise from the scientific research community to solve the issues of integrated NAMs and how to reduce, refine, and replace the animal models. And so the concept refinement and approval continued, and we received clearance in January. And after that, we started with our detailed implementation plan phase. So this is where we are now. And so we're, as I mentioned, we are very aligned with the ACD working group recommendations. And so to that end, the overarching program goals include NAMs that incorporate the following features: complex in vitro models emulating population diversity, in silico multi-scale systems simulating healthy and diseased individuals, in chemical cell-free systems capturing dynamic changes, as well as combinatorial NAMs and integrated fair data sets and AI engines for NAMs, for all NAMs. And so just a little bit more background here. So numerous activities were highlighted that were supported by NIH and other agency partners in digital twin models, in silico models, complex in vitro systems, and in chemical screening. And what complementary will aim to do is to integrate those components into the program, including in vitro and silico and chemical and data ecosystem. So really it's to provide an integrated solution that has not been possible before. And so in terms of this, just to give you more flavor for strategic planning activities, we had 3 listening sessions with major stakeholders, including academia, industry, NGO, government, and international stakeholders. And those listening sessions were summarized, and we have a summary published on our website. But we will also ensure that that information is shared with everyone. We also have we had an interagency retreat that included NIH, FDA, EPA, NSF, ARPA-H, BARDA, VA, DARPA, NIST, NASA, CPSC, and ICVAM. So we were very happy to have that meeting here at Nature last fall. And so that meeting actually, we had breakout sessions to discuss gaps and opportunities and challenges in the space, specifically from the scientific perspective as well as the regulatory perspective. So some of the highlights here are areas of priority that were discussed there, but they're just a list and not necessarily comprehensive here. It's just to highlight a few, including chronicity, neuroscience, personalized health, cross-disease pathogenesis, and population diversity. And from an operational perspective, there was a need identified for shared data infrastructure, standardized frameworks, validation, and training. We also engage in other planning activities, including the landscape analysis. So the methods paper will be published later this year. And we also just announced the winners of the Ideation Challenge Prize. So there's a QR code there, and also we can share the link to the Challenge Prize winner. So really, this was to further conduct planning activities and utilize the challenge mechanism as a unique way to crowdsource solutions for strategic planning. So normally a challenge is issued when you have a funded program. Here, we actually wanted to ask the community for ideas on how to best implement a program for NAMs and get those ideas from the community. And so we wanted to we announced this previously, but just to let this audience know about the winners here. So we selected 20 winners that each won $50,000 for their innovative solutions. And you can see the winners at this link, and we can also share it after the meeting. So we're very happy to have had a robust response to the challenge initiative. So here I'll briefly talk about the program structure for complementary. So it will involve what we're calling 3 pillars. The 3 major pillars are the top 3 bullets: technology development centers that will be used to stimulate the development of NAMs to fill in areas of greatest need. And that will the emphasis will be on increased biological complexity and throughput, as well as innovative combinatorial approaches and data sharing. Along those lines, we are also developing a NAMS data hub and coordinating center. We have a new acronym for it, so it is in our system. So this data hub will be used to integrate data structures, including standards for model credibility and improve fairness and interoperability. And one key component of this data hub will be to focus on that interoperability with other federal stakeholders and private partners to enable this interoperability planning from the very beginning, so that we're not developing something that then has to be made interoperable. And the third pillar, which is going to be a key component of the program, is the validation and qualification network. This is a validation and qualification network for NAMS adoption and implementation to establish common data elements and standardized reporting, apply validation qualification frameworks, accelerate deployment and regulatory implementation of NAMS. I will go into more detail about this in the next few slides, but essentially we are exploring a public-private partnership model to enable industry and other stakeholder in the private sector participation in this network to select use cases to further develop NAMs of greatest need to get them through the validation process into implementation and adoption of NAMs in the community. And then speaking of community, really the community engagement and training component is permeating throughout these three pillars because we appreciate the need for community engagement early, as well as training the next generation of researchers and the current generation of researchers to get them to learn more about NAMs, to develop them, and how to best utilize them in their research settings now and in the future. And so that will be used to promote the development of an inclusive, diverse biomedical research workforce with the skills to build and use NAMs, and including community engagement and societal and ethical considerations. The next slide here just shows a visual representation of what I just discussed. So these are the comprehensive centers that will include pilot projects, integrated NAM technology development, community engagement and training, and will also include core facilities, including admin, LC, data, and collection sharing. And these centers will be integrated into the consortium through a NAMS data hub and coordinating center. So we envision funding approximately 5 to 6 centers as part of this program. And as I mentioned, that will be integrated with the NAMS data hub and coordinating center. And then also the third pillar here, it's the validation and qualification network to really close the gap between developers and end users, as well as the adoption and implementation process to make sure that the milestones are well defined to enable this adoption and implementation of NAMs. And so this validation and qualification network will include ICFAM, federal partners, regulatory partners, industry partners, international nonprofits, and other stakeholders to help to develop a design phase for the public-private partnership that will then be used for the implementation phase when identifying the use cases for further development. And so along those lines, the public-private partnership here, I'm just highlighting what we are currently exploring. It's really a validation network, validation and qualification network to bring industry, NGOs, nonprofits into the network, leveraging existing FNIH public-private infrastructure, and to consider pre-competitive data sharing and potentially supporting validation activities across labs and locations for specific use cases. And again, we want to support community outreach and training throughout the process as well. And this would really provide a fluid funnel for potential solutions which may be developed through the comprehensive centers. And of course, we invite other federal partners and stakeholders to participate in this together and to synergize and coordinate with the global activities that were discussed earlier today as well. And so this is another visual showing sort of the overarching flow for this. So this diagram illustrates the key components of the complementary program, including technology development, centralized data resources, validation and qualification, with training and dissemination being the substrate that will be the key for sustainability of the program in the future, and really keeping the stakeholder engagement and implementation in mind in a bidirectional fashion. So really to provide a fluid conduit for stakeholder engagement to implementation. And the next slide. Okay, so here, just in summary, the complementary program aims to innovate, integrate, coordinate, and transform. So we aim to innovate by understanding human health and disease pathways across diverse populations using new approach methodologies, to integrate innovative NAMs in vitro, in chemical, and in silico with AI and fair data ecosystems, to coordinate with institutes and centers and agencies and public-private partnerships, and to transform the way we do basic translational and clinical sciences by leveraging the full scientific toolbox to do that. And so with that, I'd like to end my presentation and really thank you for your for listening to my presentation and take your questions.
Thank you so much, Margaret. Questions in the room? Please go ahead.
Do you have an expected timeline on when RFPs are going to be, or when the RFP is going to go in?
They can't see you, so if you could identify yourself.
Oh.
I think it's, the mic is off, I think, too.
It's on, all right. Sean McCullough, RTI, technically less than competent apparently, with microphones. Are you able to provide an approximate date on when the RFP is going to come out?
Yes, I can provide general timeline. So essentially, the concept was cleared in January with a timeline that's been adjusted. So we're likely to publish funding opportunities later this year.
Thank you.
There is a hand up from Randall.
Okay, Randy, if you could go ahead and unmute yourself to ask your question, that would be
Yeah,
fine.
yeah. Thanks for the presentation. I was just curious, are the centers going to be sort of tissue or disease focused? You showed some areas that were of need. And so I was just curious as to, for us to go ahead and start like pulling together colleagues.
So I think if I understand the question correctly, is if it will be a tissue or disease focus of the
Yeah.
program.
Like for neuroscience, would it be one center for neuroscience, or would it be one center for a particular tissue?
So I think it will be probably the former. So it will be sort of broader scope centers focused on, for instance, chronicity, neuroscience, longevity, so over the lifespan, population diversity. Within the centers, though, because we call them comprehensive centers, there'll be numerous projects as well as pilot projects that will be feeding into the centers. That being said, we envision the funding opportunity to highlight priority areas, but not necessarily be the deciding factor. So we will see what applications come in and then identify those that meet the criteria and the metrics as outlined in the funding opportunity.
Lastly, just to piggyback, is there a size of a ballpark estimate on each center, how much funding would be available?
So I can share that because that was publicly shared during our concept clearance at the Council of Councils. So the ballpark for the entire program will be 30 million per year over the course of 10 years. So that's the other thing I didn't mention is the common fund programs are unique in that they can, the duration may be 5 to 10 year time horizon. So we're very happy that that can be a possibility for the centers. Approximately, if we fund 5 centers, it would be 2 to 3 million per center.
Hi, Margaret. Charles Kovach, US EPA. Really impressive presentation and all the different activities that are being planned with so much happening. How are those activities going to be communicated? Like some of the different milestones and updates and things like that. Is the best location like, is there a website or are there planned like updates like quarterly or semi-annually, things like that?
Yes, that's a great question. So major updates are being posted and have been posted on our complimentary website, which is in the Common Fund within DPCC, so on the NIH website. So I included the link there. So major updates will be there. We will also have biannual meetings we envision for the stakeholders. And then we also have federal partners group and other stakeholder meetings on a quarterly basis. So we will be providing those updates regularly.
Great, thanks.
Hi, Andrew Rowan from Wellbeing International. I'm just curious. I mean, it's a wonderful vision, remarkable, frankly, given my past experience with NIH and the biomedical research community. What I see from this is the importance of technology, importance of new techniques. If you go back to Medawar and Medawar's description of how research advances, he says that the new techniques is really important. And I've always felt that alternatives are simply new techniques with an attitude. And that's precisely what has come out here. To what extent did people talk about the role of new techniques in science in this process? I'm just curious.
Well, just from my perspective, I think that is a very common topic that comes up. It's, high risk, high reward applications are always on the mind of a lot of the investigators that are coming in, particularly the next generation, and new techniques and strategic planning for those new techniques. What will happen next? So not just develop something to put it on a shelf, but how to actually utilize it. So there's more conversation about community outreach, community engagement in using those new techniques for the end user and not just for themselves, but for the community and the world.
Thank you so much, Margaret. Okay, and now I would like to invite our colleague from the Foundation for the NIH, Dr. Dana Connors, come up and talk to us about activities they have ongoing. Sorry, there's about 5 different iterations of how this has to go.
Looking forward to this, and then looking forward to this all day.
Let's see. Let's see how, oh, yours is playing ball. Okay.
Okay, lucky me. Right, all right. So my objective here is to contextualize the public-private partnership portion of what you've just heard. And thank you to Drs. Mizuko and Wachichinska for setting me up so nicely, and to Dr. Kleinstroyer and ICFAM for having me. So I'm from the Foundation for the National Institutes of Health. I know how to do this. Excellent. We are a 501c3 nonprofit that was founded by Congress in 1990 to support the mission of the NIH. So that largely is like the Reagan-Udall Foundation. There are other foundations that are set up to support government agencies and to serve as a way to engage industry in particular, but also to raise funds and do activities that the agency can't do with their own congressionally appropriated money. So a really impactful but small example is we helped raise the funds to build the Saffir Lodge for patients who live here on campus and are on trial, and before then didn't really have a place to stay. And so that's been a really impactful and great thing that we did. Our bread and butter is largely about developing public-private partnerships. That's our main effort. So we work across all 27 institutes. We're a relatively small organization where we just hit 100 people. We've quadrupled in the last 5 years or so. And we work across all 27 institutes to coordinate with industry as well as other agencies. We work very closely with FDA as well when it comes to qualifications. And in the last 20 years or so, we've raised over $1.5 billion. We're very proud of this effort, and 90% of those 90 cents on dollar goes directly to science and not to our overhead. So we're very happy about that. We have multiple metrics that shows that we do good things, and I'll let you do that. I'm sure these slides will be made available. You can read those at your leisure. But largely what we do is figure out the governance of a PPP, a public-private consortium. We develop the policies that will govern that, including how IP is going to be handled, how publication will be handled, conflict of interest, confidentiality, those kind of things. And we implement those across the range of stakeholders, essentially providing a playing field on which all stakeholders can come and play equally. We also are largely an organization of project managers and program managers. So we stand up and then run those programs and close them down, hopefully, if all goes well. And then we also do the fundraising and relationship management that's required to build them. We do it in 3 main ways. And so I'm going to give a couple examples of these and then try and describe how we can apply this to complementary, which you just heard about. The first is essentially the NIH or the White House comes to us and says, we've got a crisis, solve it. And they just give us money to do it. It's quite rare. We did it with the opioids epidemic. We addressed opioid therapeutic development. And then also with COVID, we were largely responsible for standing up and managing a lot of the vaccine trials and therapeutic development. What's much more common is this middle column where we build a partnership in coordination between the agencies and private stakeholders. And we try to fund those equally. And so I'll describe a little bit of that. And then the third model, I'll use Biomarkers Consortium as a example, is when we build a program and then we basically pitch it to industry. And it's 100% funded by private dollars. So as an example of that middle column, the Accelerated Medicines Partnership was started in 2014 to build long-term, large data-driven discovery efforts for therapeutic development and diagnostic development, as well as biomarker development. We've done this in a range of diseases: Alzheimer's disease, type 2 diabetes, Parkinson's disease. And actually, I'm happy to say that just today, I received on my phone an announcement that AMP ALS was launched, which will be the largest data hub for ALS data yet. And the objective there is really in diagnostic and therapeutic development and design. So we're kind of continuing this. There are 9 of these now. Wow, okay. This isn't my slide. That was exciting. So I hope you enjoyed it. Through those 9 partnerships, we've raised about over $700 million, and it goes to a range of institutes. And including industry, the requisite agencies, and then also nonprofits that research institutions, whatever's needed to kind of get the job done. And here's some examples of the partnerships themselves, how the consortia came together. So in Alzheimer's, a range of pharmaceutical companies that are engaged in that space, the requisite institutes from NIH, and then the nonprofit members that really are needed there for subject matter expertise, as well as community support. And so we did that across a range of diseases. The Biomarkers Consortium, by contrast, is a much smaller consortium type in that it tries to encourage shorter timeframe projects, smaller funding, and faster movement for validation and qualification of biomarkers. So we work very closely with the biomarker qualification program at FDA to identify biomarkers that we already know about, that have a discrete context of use, and then to move those a little faster down the pathway for validation. And really the objective here is just to get drugs approved and developed faster, and to build drug development tools that we can really use to help patients in the end. We've done this for, well, I guess we're coming up on our 20-year anniversary now, so this slide's a little out of date. But in that time, we've qualified some biomarkers, publications, citations, et cetera. But we work very closely with FDA on guidance documents as well. And we have now over 60 member organizations. And this is kind of how we fund the Biomarkers Consortium is we have a tiered membership profile based upon R&D budget. Companies can come in, pay a membership fee to sit on one or multiple steering committees. For a long time, I ran the Cancer Steering Committee, and I had counterparts in inflammation and immunity, metabolics, and neuroscience. And all of them work collectively to sit under what is an executive committee that evaluates those and makes sure that we're aligned with the strategic vision of the Biomarkers Consortium as it was construed. Doug Lowy, 3-4-time acting director of NCI, is now the co-chair for the executive committee, so we're working very closely. Actually, this Friday, we'll have our executive committee meeting, and we'll evaluate and hopefully approve 4 new projects for launch. 3 new projects for launch. One of them is concept, so there's kind of a concept project approval process that happens. But you can see here that we try to really benefit from the engagement of some high-level stakeholders from across the NIH institutes, across the FDA centers, as well as from industry and nonprofit organizations, as well as our own board. And the way that it's set up is that executive committee that you just saw sits over the top of these 4 steering committees that I mentioned. And then each one of those steering committees works to develop working groups that can bring concepts to approval and put those forward through our approval process. It's kind of a concept development, concept approval, develop a plan, approve the plan, launch the plan, move on the plan, close the plan. That's the basic brushstrokes of how this happens. So for complementary, I'm hopeful that not only can we capitalize upon these existing consortium models that we have, and I will frankly say that no 2 consortia that we build are the same, right? They are always unique, they're always different, and they're always challenging, but they're usually rewarding. But I think we have some good models like AMP and like the BC that we can use for complementary as well. So the vision, working with Nicole and Margaret and others, for a PPP in NAMS is really that we can create one that provides for a community platform and replication of processes that will engage, Andrew, to your question, novel technologies as well as existing technologies, and engage stakeholders from across the validation process. And then take those recommendations and be able to put them into public guidance and support the existing work that's going on. I think it's really important to note that none of us want to remake any wheels here, right? We've heard over the last 2 days a lot of great tools and a whole lot of great resources that we can put into action here. And hopefully we can just support that momentum without redesigning anything crazy. So where we are right now is what I would call a pre-design phase, while the Office of the Director and Office of General Counsel and Office of Science Policy is reviewing a proposal that's being put together for collaboration between the NIH and FNIH to put this together. So as Margaret mentioned, some landscaping is taking place, engaging with stakeholders like yourselves. So my pitch, if you're interested, if this is perked your ears and you're interested in becoming part of the validation and qualification network, or you're interested in talking to me about this PPP, I'm certainly available to you. And then once we have that approval, we can establish the PPP itself. The first phase would be in implementation, would be years 2 to 5, as it were. As Dr. Chichinska mentioned, this is a 10-year program, so if we can do phase 1 in 5 years and then a second phase in 5 years to follow on is kind of how we envision it. But that first one would be kind of a proof of concept, right? That we can establish success criteria, the minimum information required from NAMs, develop what we mean by validation and qualification, and then put in a governance structure that can manage that, similar to what I showed from the Biomarkers Consortium, whether that's an executive committee sitting over multiple steering committees that guide in silico models, in vitro models, and in chemical models, or if we want to do it on a disease basis, I think that should be determined by the network when it comes together. But we certainly want to identify those important stakeholders that need to be at the table, have frequent conversations with them, and seek their feedback in figuring out what the scope is going to be for that design. And then in the actual implementation phase, we would select several case studies that we could put through this validation process. Likely, these will be established NAMs that we can use to design the validation process, with the objective that we could then incorporate novel technologies in the future. Obviously, establish that data sharing process that we talked about through the data sharing hub. And then using the RFP process and the prize winners that Margaret mentioned as well, bringing on additional participation. Oh, and I would be remiss if I don't mention that I envision that in addition to the validation and qualification network that Margaret mentioned, I think the other great piece that FNIH can at least coordinate here is that education and training piece. I think that's going to be really critical to making sure that these are implemented in a broad and open manner. And so I'm open to input on how we engage that as well. So I think the FNIH in particular can kind of pull these stakeholders together, do what we do best as far as program development, coordination, administration, et cetera, and then the management of it as well. We'll have to see, obviously the prize winners were just announced, so that's great. We've got to start going, but if we're going to do additional awards, we can help manage those, informing peer review processes, and then, of course, providing oversight for financial management, follow-up, et cetera. And then working on that education and training piece with communications and support. And then providing the governance that's needed to put all this into place, of course. And I envision that industry will have just as important of a role as the agency does. And they can not only bring financial support to the program, but technologies, subject matter expertise, other investments that we can really rely on. And the FNIH model, the even playing field that we like to build, is based upon that input of information, creating a system of equity, right? We may not bring in the exact same things as far as subject matter expertise or as far as funds go, but we do our best to create a sense of equity so that everyone is collectively at the same table. And yeah, with that, feel free to contact me if this piques your interest, if you're interested in the validation and qualification network. I envision that we'll be in the next weeks, hopefully not months. We'll be seeing the RFC come to the FNIH board and we'll approve it, and then we'll be pulling together the initial network meeting to build our design. And happy to take questions.
Thank you so much, Dana. Questions in the room? Yep, please use the mic and please identify yourself.
Hi, I'm Chantel Nicholas from App Global. Quick question. Thank you for your presentation. I was just wondering, has industry demonstrated a high level of interest in development and use of NAMs? Or is that, are we engaging in finding that out?
Yeah, great question. Thank you. I probably should have mentioned this. So before actually I heard about complementary, I was really excited about NAMs myself. And so I started, and I was working in cancer. And so I was just reaching out to all the pharmaceutical companies that I know, like all the big players, and saying, hey, can you put me in touch with someone who's working in this space? I want to know what's going on. And I was overwhelmed by the enthusiasm and response from the people I talked to. There's a lot of interest in building a program like this, taking part in it, the acceleration that's needed. In fact, I would say that the overarching message that I heard from large pharma was, we are working furiously to implement NAMs, but they require internal validation. And I know that all of my competitors are doing the exact same thing, and we're duplicating efforts. And I can't do it fast enough because the technology is moving so fast. This is a prime and ripe place for public-private partnership. And then at the same time, talking to the technology developers, and they're saying, we just want to get these in the hands of the people that can use them, but we also need the support of a public-private partnership to bring this together. So I'm really enthused that everyone on the industry side that I talk to is just on board. And I think this could come together really quickly based upon that initial data. I don't know if is Randy's hand still up, or is it?
Let's check. Randy, is that a legacy hand or oh, there, it went away. Okay. Legacy hand. No online and no other questions in the room. Okay, fantastic. Now you all understand why I was so excited about this session. So I just want to say just a huge thank you to Anna and Margaret and Dana for spending time with us here today. And I think we will now take a break for lunch, and we will reconvene at 1:20 p.m. So that's just over an hour. I double checked that math before I said it. And thank you so much, and we'll see you back here in an hour.